Yes, welcome everybody to the second day of the Brain Prize meeting. It's a great pleasure to be chairing this second day. Today the topic is genomics, genetics, identification of mutations. And we have a great lineup of speakers. So the afternoon will be articulated like yesterday. So there will be 3 25-minute talks. So one talk by Matthew Hurls from the Wellcome Sanger Institute, followed by 10-minute questions and answers. Then there will be a 25-minute talk by Mark Daly from the Institute of Molecular Medicine Finland in Helsinki, from the University of Helsinki, again followed by 10 minutes questions. Then we will have 3 flash talks, one after the other. So flash talks of about 5 minutes by Daniel Calame from Baylor College, by Stamatina Tsanuligou from the University of Lausanne, and by Andrea Asenjo Martinez from the University of Copenhagen. And then there will be a question and answer session after these 3 flash talks. After the Q&A session on the flash talks, we'll have another 25-minute talk plus 10-minute question and answer by Joseph Buxbaum from the Icahn School of Medicine at Mount Sinai. And there is a change in schedule, as yesterday Jonathan Mill could not give his talk due to technical problems. We managed to convince Jonathan to give his talk today. Unfortunately, we have to replace the panel discussion by the talk, so we won't have the panel discussion at the end of the day, but we'll have Jonathan's talk. I think it will be an extremely nice way to finish the afternoon, because he is working really at the interface between the genome and the epigenome and the interactions and crosstalk in the context of neuropsychiatric diseases. So I think it's really great that Jonathan will ultimately be able to give his talk. So that will be a 25-minute talk plus 10-minute Q&A. So his talk will be about epigenome trajectories in neuropsychiatric diseases. So like yesterday, you are all welcome to ask questions using the window on the right. So you can ask your question during the talks, and people can also rank the question. If you like the question that people ask, you can vote for these questions, and so it allows you to have a ranking of the most preferred question that may help the discussion. So I really invite every one of you to ask the questions along the seminars. Since we won't have the panel discussion, I would like to suggest that we, at the end of each 25-minute talk, that we spend a little bit of time to allow the speakers to give their opinion about the question of where do you think genetics will be in 10 years, and what do you think will be your contribution to this? So a little bit on the format of yesterday to make sure that we also have the opinion on this perspective for the field. Okay, so without further ado, I will let the stage to Matthew Hurls from the Wellcome Sanger Institute in the UK, and his talk is about the genetic architecture of neurodevelopmental disorders. Matthew.
Thank you very much for the kind introduction, and I see that my video has just frozen, but rest assured I'm still here. It's a great pleasure to present at this meeting, and doubly so because of this meeting being dedicated to the fantastic work of Adrian Bird and Huda Zoghbi on Rett syndrome. And it is connected. I'm afraid it's frozen, so maybe we could just go ahead. I think my face is the least important part of this. So to recap, it's a great pleasure to give this talk at this meeting, recognizing the fantastic achievements of Huda and Adrian. And I'm going to be talking very much about the genetics and not focusing on the kind of downstream mechanisms, or indeed the kind of the therapeutic kind of potential. kind of recognizing that there are other, experts talking about this at this meeting. And I'm going to be trying to focus not just on what we know now, but what we can tell about the overall genetic architecture. And by that, I mean the overall contribution of different types of genetic variation to neurodevelopmental diseases, even if we can't necessarily pin down particular genes in that. But they certainly give us a clue where to look next, and potentially some ability to, as Isabel nicely put it, to predict where we might be in 10 years' time. So I'm going to be talking largely about our work in the context of a UK-wide study called Deciphering Developmental Disorders, which has been going for about 10 years now, and is very much a collaborative effort between families with children with severe rare developmental disorders in the UK and the clinical teams within our National Health Service who care for them. And there's 24 centres across the UK and Ireland that participate, shown as the black dots in the map on the right. And the project really started with two principal aims. The first, to understand what the genetic basis might be, so a classic research kind of aim, but also a translational aim to think about how we can use the new genomic technologies that we as a research institute had access to in advance of our national health system, and how we might use those technologies to improve diagnosis. And I'm going to be focusing very much on sequencing of all of the genes in the genome, so-called exome sequencing. So over the course of about 4 years, over 200 clinical geneticists in the UK caring for these families recruited over 13,500 families, most of those in the context of being able to have DNA from both the child and both parents, which crucially allows us to identify genetic variants that are newly arising in the child, which I'll talk most about during this talk. The clinicians also systematically characterized the clinical phenotype. In this research study, we were also trying to have positive benefits for the family and feeding back likely diagnoses to the clinical teams. And as I'll mention, we can achieve about 35% of the families we can get a concrete genetic cause for their disease currently. So the nature of these children is that they're quite varied in terms of their phenotypic presentation, and that's deliberate because they're representative of this clinical population who need a diagnosis. But by far the most common presentation is that of neurodevelopmental disorders. And I've listed some kind of broad summary statistics here in terms of the numbers that have intellectual disabilities, seizures, abnormal brain structures revealed by MRI, or indeed autistic features, although this is almost certainly an underestimate in terms of the proportion with autistic features. But also there are many other disorders of different systems manifesting, and many of these children have multi-system disorders. And we track information about how the child's developed over time, and of course the family history. And in most cases, these children are the only poorly member of their family, the only affected member of their family, and everyone else is healthy. That's the most common family history that we see. So going into this study, we thought about the different possible contributions to the etiology of these disorders, from purely environmental causes, gene bioenvironmental causes, kind of at the top here, to classically Mendelian disorders being recessive or dominant that might be in protein coding genes or outside of those genes. But also the potential for contributions of combinations of variants, be they oligogenic or combinations of many hundreds or thousands of variants, and so polygenic. And so what I'll show you throughout this talk is some kind of plots that look a little bit like this, where we're trying to look at the proportion of individuals who have that underlying genetic cause as part of their the root cause of their syndrome. And so going into this study, one might have anticipated that given the severity and the rarity of these disorders, we would find that the vast majority of children had a Mendelian disorder of variants in protein coding genes. But equally, we might be looking at a situation where only a minority have those kind of causes, shown by the kind of green bars at the bottom. And actually, there's a large contribution from these combinations of variants, either polygenic or oligogenic. So quite an open mind, but we really want to kind of build this picture up over time. So the first place to look is what are the diagnoses that we can make, given that we can make diagnoses in about a third of these families. And one point to make is that we currently, the kind of diagnostic gene list that the clinical geneticists have put together for us to use comprises just under 2,000 genes. So that's about 10% of all genes in the genome. And it goes to underscore one of, I think, the key challenges in neurodevelopmental disorders, which is that our ability to discover new disorders in a robust way such that they can use to be diagnosis is running far ahead of our ability to characterize the biological mechanisms or indeed develop the therapies for these disorders. And one would hope that Rett syndrome provides us with something of a template about how we might progress with some of these disorders that we've only discovered more recently. So if we look at the diagnoses that we make, and here I'm showing you the top 20 diagnoses that we made in this cohort, the first thing to note is that none of these conditions accounts for more than 1% of all of the children. So there's a very long tail, a very heterogeneous set of causes in these children. The second is that all of these 20 most common disorders are all dominant disorders where the majority of pathogenic variants are de novo mutations that have arisen newly in the child, not present in either parent. And the sharp-eyed among you will probably see that MECP2 is on that list of genes at the bottom in this table, in about kind of 13th place in terms of the most prevalent disorder. So it's one of the most prevalent disorders amongst this set of disorders. The other thing one can see is that the majority of variants in these most common disorders are having a predicted to truncate the protein, so-called protein truncating variants. So most of these genes are haploinsufficient disorders where one copy simply isn't enough for normal neuronal development. So how do you go about, given that that's a suggestion that maybe the place to look for new disorders, which is going to be most powerful, is to look at de novo mutations and to look for new genes that haven't previously been associated with developmental disorders that have an enrichment of de novo mutations compared to what one might expect by chance. And here we use our knowledge of the germline mutation process and how mutable every single base is in the genome. And this was work done by Caitlin Samoka when she was in Mark Daly's group. And we can use that to estimate the mutation rate of every single base change in a gene, and we can predict the consequence of those base changes. And so on a gene-by-gene basis, we can build up a picture of how frequently should we expect to see a de novo missense change or a de novo protein truncating change. And we can do this on a gene-specific basis. And then we can detect those de novo mutations in these large cohorts of heterogeneous patients and simply ask the question, do we see more of these different types of damaging variant than we expect by chance? And so here I'm showing you a cartoon version of 3 genes where I'm writing down the expected number of de novo mutations we might expect to see purely by chance due to this underlying mutational process. And we can compare those expectations to what we see in our patient cohorts. And so most of the time we see a picture very similar to what we see for gene 3 on this cartoon, which is the number of mutations we see in the patient cohort is entirely consistent with these just being randomly generated mutations by chance. Whereas sometimes we see in gene 1 that there's an excess of protein truncating variants. And sometimes we see as gene 2, there's an excess of missense mutations. And so we've used this approach, as have others, and you may well hear about that in subsequent kind of talks, to identify genes where there's very strong evidence of association with neurodevelopmental disorders. And this is showing you 14 such genes that we published a few years ago, and showing you the primary protein structure of the proteins that are encoded by those genes. And so I don't expect you to read all the details, but the key thing to note here is there are 2 major patterns. The first is that, as shown here, 4 genes where the mutations which are shown as red lines above each protein are all missense changes, and they're all clustered in one particular part of the protein, as compared to these other 10 genes where there's a mixture of missense and truncating variants that are spread throughout the gene. And we think this is giving us a clue into the underlying mechanism, whereby haploinsufficient genes tend to show this pattern of a mixture of truncating and missense variants spread throughout the gene, whereas other mechanisms activating gain-of-function mechanisms tend to be missense variants clustering in one particular part of the gene. So we're getting some clues into the mechanism of disease, as well as identifying novel genes associated with neurodevelopmental disorders. So that suggests that we can A, identify diagnoses in these children, B, we can identify new disorders where there's sufficient evidence in any one gene, but it doesn't tell us necessarily what we haven't yet discovered. And there are ways in which we can characterize that by instead of looking at individual genes and asking how many mutations are there compared to what we would expect by chance, we can look across all of the genes, the entire exome, all 20,000 or so genes, and simply look at the overall burden of different classes of variation, and to give us an expectation of what is the overall contribution of that type of variation. And so the easiest way to think about this is a kind of a case control type analysis, where we might look in our patients and, for example, see how many damaging de novo variants we might see in 10,000 patients, compared that to 10,000 children who don't have developmental disorders, and see what fraction of those have the same types of variants. Now, sometimes we don't always, and the difference between the two is what we call the burden or the excess. But sometimes we don't have those control data, and we don't have 10,000 control trios quite yet, although we may do soon. So sometimes we use a population genetic model, and that's kind of an example of what I was showing you before, where we're using our understanding of the germline mutation process to build up an expectation of what we should expect to see by chance. And when we do that in the DDD cohort, we see this kind of picture, where we see that in red, there's a 2.5-fold excess of truncating variants and a 1.5-fold excess of missense variants across the cohort, compared to synonymous variants where there's no excess of de novo mutations at all. And if one sums up that excess across the missense and the truncating variants, and one considers that we've looked at about just over 4,000 families here, then it gives us an estimate that just under half of the cohort have a significant contribution from a pathogenic de novo single nucleotide variant or an insertion deletion. We can also start to build up a picture of the relative contribution of haploinsufficient disorders and so-called altered function disorders, because we can look at known haploinsufficient disorders or HI disorders. And when we look at those known disorders, we see roughly a 1-to-1 ratio of truncating and missense variants. Whereas we look at disorders which we know are by an activating or dominant negative mechanism, we can see there's a huge preponderance of missense variants. And if we apply that to the absolute numbers of excess variants that you see on the left-hand side, that gives us a rough estimate that this overall burden of mutation is roughly 60% loss of function, 40% some form of altered function. So when we apply that kind of thinking to that overall burden of just under half of the cohort having a de novo coding variant at the root cause of their disorder, we only about half the time is that variant landing in a gene that we've already associated robustly with neurodevelopmental disorders. About half the time, half of that burden is as yet unexplained, and we think that roughly half of that remaining burden is going to be more haploinsufficient disorders, and roughly half of that is going to be altered function disorders. So we have a sense of a substantial fraction of the undiagnosed patients in the cohort we have actually detected the variant that's causing the disorder. We just haven't yet gathered enough evidence to associate robustly that gene with neurodevelopmental disorders. Now, we can apply that same kind of approach of assessing the burden across all genes to other classes of variation. And this is I don't have the time to talk to you about all of those, but this is the kind of picture that we built up here, which is then in an outbred population such as the UK, then by far the biggest chunk of that genetic architecture of these kinds of classical Mendelian type disorders is coming from dominant mutations in protein coding regions, this kind of big green box at the bottom, with smaller contributions from X-linked disorders and recessive disorders. One kind of key point to note is that there is some significant heterogeneity, both globally and within the UK population. And the picture here just showing you the balance of dominant disorders versus recessive disorders in the British European ancestry component within the DDD cohort is very different from that in the British Pakistani ancestry component of the DDD cohort, where but this is driven not so much by the actual continental ancestry, but by the practice of consanguinity and parental relatedness. If one stratifies the British Pakistani subset into those whose parents are related and those where the parents are not, those where the parents are not show a very similar kind of overall architecture to the British European ancestry. So it's predominantly about parental relatedness rather than continental ancestry. But you can see here the contribution of recessive and dominant disorders is more like 1-to-1 as opposed to 10-to-1 in the British European ancestry component. So there is significant heterogeneity across populations in the world and in the UK. We can use this kind of approach to contrast and compare different types of neurodevelopmental disorders and different types of rare disorders where we think rare variants might play a role. And here I'm showing an example of an analysis we did with comparing the DDD cohort, which is all postnatal, with a prenatal study where we were sequencing trios using exome sequencing, parent-fetus trios, where an ultrasound abnormality had been picked up, and quite diverse, sometimes cardiac, sometimes brain, sometimes multi-system, sometimes skeletal. But what one can see from this is that the overall contribution of de novo mutations is much higher in the DDD cohort than it was in the PAGE cohort. And one of the reasons why the diagnostic yield that we were seeing in the prenatal cohort was much lower is not so much that there are many new disorders that are only observed prenatally that we have yet to discover, and more that actually classical Mendelian disorders are less common in this population, and we need to look for other causes. So going back to this, the picture that I showed you, there's a big grey box where we don't actually know what the genetic architecture is. And one of the components of this is the potential contribution of polygenic inheritance. And here's some lovely work by Mari Niemi, Hilary Martin, and Jeff Barrett has shown that there's taking the kind of approaches that are commonly used in genome-wide association studies for common disease, we're able to show there's significant heritability due to common SNPs for being present in the DDD cohort, that there aren't individual SNPs that are common SNPs that are genome-wide significant, but that the overall risk alleles are over-transmitted from parents to children, as one might anticipate and as has been seen for autism. And when one looks at this kind of genetic architecture of common variants and compares it to risk variants seen among common variants for other traits, there's a clear correlation with those cognitive-related traits that have been studied, such as educational attainment, measures of intelligence, and schizophrenia risk, in the directions that one would expect. In other words, increased risk of being in the DDD cohort is positively correlated with increased risk of having schizophrenia, but also with the likelihood of having fewer years of schooling and lower performance on intelligence tests. Whereas there isn't strong correlations with other kind of metrics, other GWAS summary statistics shown below. So one of the questions was, well, is that polygenic risk really only found in that grey box, those undiagnosed patients, or is it actually spread more equally throughout the cohort? And we couldn't see, although we don't have complete power, we couldn't see that it was strongly stratified between the undiagnosed and diagnosed patients. Indeed, we could see that this polygenic contribution is influencing the clinical presentation. And the evidence for this is we can take polygenic scores that have been generated in the general population for traits like birth weight and height, or intracranial volume, educational attainment, autism, and we can see whether those polygenic scores influence the probability that a patient in the DDD cohort has been either annotated with those features or the quantitative measurements that have been taken. And we can see there's a strong correlation with all of these with the presentation. So certainly this polygenic component is influencing how these diseases are expressed, even if one has a strong rare variant that's driving a lot of the causation. So the evidence I've shown you before suggests that if we want to get much further, then one route is to understand these dominant de novo disorders that we haven't yet discovered. And to do that, we're going to need larger sample sizes. And so we just recently worked together with a large diagnostic company in the US called GeneDx and a leading diagnostic center in the Netherlands at Radboud University. To combine the data on similar cohorts of patients with largely neurodevelopmental disorders. And here the analysis, overall analysis is just over 31,000 trios. And what we found, and this is just the one piece of kind of with minimal functional information that I want to show you, is if we look at the top 100 most prevalent disorders in this combined cohort, so this is a blown out version of what I showed you previously for the top 20 disorders, and then broadly look at the different functional classes of these genes, we see there's two major functional classes, those that involve chromatin genes, those that involve synaptic genes, and then the remainder. And on this list, actually, MECP2, although you probably can't see it, is actually the 5th ranked gene in terms of most common disorders. So we were able to discover strong evidence for 28 novel neurodevelopmental disorders, some evidence coming in blue from that kind of haploinsufficient kind of pattern of mutations, some evidence coming in red from that kind of altered function pattern of mutation. But the glass half full is that we discovered 28 novel disorders, and that leading to the diagnosis of a few hundred patients who weren't previously diagnosed in those cohorts. However, the orange bar here shows you that we've only just nibbled away at this excess of de novo mutations that we know exists in this cohort. So even with a sample size of this scale, we have still yet much more to discover. So one of the things that we did observe, and maybe of interest to you, is that there's if one looks just purely at the autosomal genes, there is an increased burden of de novo mutations in girls than boys. So it points to some important sex differences in architecture, in the genetic architecture. But we also need to think about why we haven't discovered many of these novel disorders despite the sample size. And there's three major kind of reasons that we've been thinking about. The first is the disorder has a small mutational target. It's very low prevalence, and so we see very few cases even in large cohorts. That's because the gene is small, or perhaps only a very small proportion of variants in the gene can cause the disease. It might be that there are disorders that we're just seeing very rarely postnatally because there's a high rate of prenatal loss. But we think one major reason is because the mutations are not completely penetrant, and that many carriers of de novo mutations of these genes will not be as severely affected as the children in the cohort. And so the things that we need to do to address these issues are we need larger sample sizes. We need to be getting to studies of the scale of 100,000, our modeling suggests, or more. And we really need to combine data from both inherited variants and de novo mutations, and also across disease studies, because if mutations are incompletely penetrant, that means that there are many people in the general population walking around with these variants who maybe have their various cognitive or neurodevelopmental traits mildly impacted by these genetic variation. And in large enough numbers, we can start to aggregate that information together. So I don't have time to show you the evidence that we've seen that these inherited variants really are playing a role, but we can certainly see that they are over-transmitted from DDD parents to children, from unaffected parents to affected children. And we can see in the general population that these kinds of variants that we often see in children with neurodevelopmental disorders that we think are causal, when we observe in the general population, they're often having a big impact on cognitive traits. And so I'll skip over the next two slides, which basically presented evidence for those two things, to really give you what I think is what we're going to spend a lot of time over the next few years trying to achieve, which is combining evidence for individual disorders from both de novo mutations that we see in patient cohorts, from inherited variants, rare variants that get over-transmitted from parents to affected children in these patient cohorts, and quantums of evidence from genetic variation that we see in population cohorts. And I anticipate that in 10 years' time, we'll have a much better picture that will join up the rare, completely penetrant variants that we spent a lot of time focusing on to date, with the polygenic perspective that I showed you is clearly playing a role in the neurodevelopmental disorders we see in the DDD cohort, but other disorders. And that will fill in the gap between those two. And in so doing, we'll get a much better understanding of incomplete penetrance and indeed of the full range of phenotypic severity that comes from carrying some of these damaging classes of genetic variants. And with that kind of prediction of the future, it just remains for me to thank the key people who have done this work. A lot of the work I described on the 31,000 trio analysis was done by Joanna Kaplanis and Caitlin Samoka, and wouldn't be possible without our colleagues at GeneDx and in Nijmegen. And the work that I didn't have a chance to describe on the UK Biobank was done by Eugene Gardner. But with that, I'll be very happy to take any questions or comments that you might have, and apologies for seeing my frozen webcam for the last 25 minutes.
Very good. Many thanks, Matthew, for this extremely clear talk. And thank you also for sharing your perspective for the next 10 years. We discussed this already yesterday in the context of the epigenome. And unlike the epigenome, the genome is much simpler, right? There is one code. So I was wondering, with this in this perspective, the question really is, we know the genome sequence since about 20 years, and the technology around genome sequencing has been amazing, amazingly efficient, and it improved a lot in the 20 years. But I wonder, compared to the epigenome, where the epigenome is very complex, the profile, the mapping of DNA methylation, the mapping of histone modifications. There are dozens of histone modifications. There is even a histone code. So the non-coding RNA, all these are extremely complex, a lot more complex than the genetic code itself. And when you see the progress which has been done in the past 20 years since the sequencing of the genome, I wonder what were really the hurdle the slow progress in a way, despite the major effort, major funding, and compared to the epigenome, the relative simplicity of the genetic code.
So that's a really interesting question. There's multiple parts to that. We have a particular interest in these chromatin disorders, which are interfering with many of the aspects of the epigenetic code that you mentioned. And I think there's different angles in which epigenetics comes to play a role. So I think one of the things that we and others have done is show that the use of epigenetic assays can be really informative in actually making robust genetic diagnoses. So many times when you have a patient with a variant in a chromatin-related gene, you may not be clear on whether it's pathogenic or not, but it may leave a characteristic epigenetic signature in an accessible tissue. And in fact, many of the neurodevelopmental disorders caused by mutations in chromatin machinery have a characteristic DNA methylation signature. And so there's an opportunity, rather than seeing this as a kind of epigenetic versus genetic, actually the two kind of work very much in concert in that context. And when it comes to the kind of other aspect in which many people think of epigenetics, which is how does one incorporate the environment, and environmental exposures, especially in utero exposures. And I think, again, it probably is important for us to think not in a kind of genetic versus epigenetic perspective, because one can start to get pictures or predictions of the in utero environment using genetics as well as outcomes from the in utero environment using epigenetics. So, for example, we know that kind of glucose metabolism, and maternal diabetes is a risk factor for many developmental disorders. Now, obviously, diabetes there's to some degree one can predict glucose levels and risk of diabetes from genetic evidence, and therefore one can start to use genetic windows into the in utero environment, which is incredibly difficult to analyze. So I think we should be thinking about this very much as how do genetics and epigenetics reinforce one another, rather than how much do we deploy into one versus the other.
Yeah, I agree. Yeah. Okay, very good. Thank you. We have a question from Huda Zoghbi here. Thank you, Matt, for a great talk and data. Have you encountered loss of function mutations that sometimes show up in an apparently asymptomatic parent? If yes, how often? And also, have you observed some mosaic mutations in Provence, and are these more enriched in particular genes?
So yeah, two very good questions. So the second one first. So yes, we can detect of the de novo mutations we detect in children, about 4% look like they're mosaic in the child. So one thing we did was to take every single loss of function variant that we identified in a de novo mutation we identified in the child, and then sequence it, tens of thousands of fold depth to look at this, and so we can fairly concretely distinguish the mosaic from the non-mosaic. So that 4% kind of proportion of post-zygotic mutations in the child seems to hold up across other studies as well. But we can only pretty much see down to where the variant is maybe at 20% rather than 50%, as one would expect for a kind of constitutive variant. We don't have really good window of what happens below 20% mosaicism. And the first question in terms of the inherited loss of function variants, we do see inherited loss of function variants from parents to children. And we've been doing some work in some of those studies. So David Fitzpatrick's been leading analysis and anchoring D11, for example, on a more detailed phenotypic follow-up to try and ascertain, is the parent really unaffected or are they mildly affected? One of the issues that we have is you often have more phenotypic data on the mother than the father. And if we look at where we see inherited loss of function variants, we often see them enriched in the father. And yet we have poorer phenotypic information there. So I think one of the challenges that we need to face as a field is how do we get better at phenotyping parents and understanding the difference between true resilience versus phenotypic severity. And I think that really requires us to think a little bit outside the box about how we as researchers interact and work with families, because I think a lot of times families would be willing to provide this information, but it's not traditionally captured by the clinical systems, at least the ones that we work with. So I think there's some opportunities there, and I think the data is pushing us towards that direction of really understanding parental phenotypes much better.
Okay, very good. Thank you. We have a question from Anders Björklund. Great talk, thanks. Could you elaborate on the potential role of variation outside the exome?
So we did one study, which we published a couple of years ago, where we focused on the most conserved non-coding elements, and we could see a subtle but significant enrichment of de novo mutations in the most conserved fetal brain active regulatory elements. And we haven't published it. We've since replicated that in an independent cohort of whole genome sequenced individuals. But it's pretty modest. And we at the time, we estimated that maybe 1 to 3% of patients would have a de novo mutation in one of these ultra-conserved fetal brain active elements. And that holds up with our second round analysis, where we've been able to look at more of the genome. We haven't, however, looked at recessive possible causes in non-coding regions, and we haven't also really factored in kind of structural variation in non-coding regions in a systematic way. So I think there are still avenues to pursue there, but they don't look as strong to us, at least yet, as following up on the protein coding de novo mutations and the inherited variants, where we think they can account for tens of percents of the undiagnosed patients.
Good, many thanks. There are a few more questions, but for the sake of time, I think we'll not answer them directly. I invite people, I don't know, maybe Matthew, we can share this question with you, and you can answer send answers to the people who asked them. But in any case, many thanks for your talk again. And we move to the next presenter, who is Mark Daly from the Institute for Molecular Medicine, Finland, University of Helsinki. And he's going to tell us about the progress in the genetics of schizophrenia. Thank you.
Thank you, Isabel. Nice to be here, and only sorry it's not in person, because it would be much better to be in Copenhagen with all of you. I'm going to sort of continue many of the themes that Matt has raised in his excellent talk, and demonstrate the applicability and similarities and differences when we apply the same type of comprehensive approach to genetics in the example of schizophrenia. So one, obvious and important reason that we, focus on schizophrenia in terms of genetics is that despite, a remarkable, appreciation of how important and devastating and common schizophrenia is, there has been next to no progress in the introduction of new medicines for schizophrenia, other than, variants on chemicals that were originally discovered more than 50 years ago in unethical, experimentation on humans in institutions. And the fact that little progress has been made in developing novel mechanism therapies for psychiatric disorders, I think, has a component of that comes from the fact that we don't have a deep understanding of the biological root causes of disease. So what we have known, again, for many, many decades, is that schizophrenia, autism, many other psychiatric disorders and neuropsychiatric disorders are among the most heritable clinical entities that we study. And distinct from the severe intellectual disabilities that Matt was discussing, the general clinical entities of schizophrenia and autism in particular are highly, highly heritable. They run in families, suggesting that inherited variation, or at least DNA variation that you're born with, is playing a major role in predisposing individuals to risk. Now, despite that, we all lived through, years and years efforts in studying these and many other complex human diseases, in which we made next to no progress in the discovery of underlying genes, while our much more successful colleagues who had sensibly applied themselves to more Mendelian scenarios in which the genetic inheritance tracked very nicely according to Mendel's laws, were having a great success in the 1990s. So as we finally were emerging out of the sequencing of the human genome and developing technologies to approach genetics and in a much larger scale than previously had been, we had the ability to do, we, as a community, entered into a period of extensive debate over what approaches should we take to tackling the obviously highly heritable but much more complex and non-Mendelian common phenotypes that were of high medical interest and burden to society. And so often these debates were rather, fruitless, debates about, just one's prior opinion, since there was very little data at the time, but generally came down to a debate about whether we should look at common inherited variation in which there might be weak and perhaps regulatory risk factors, or whether we should focus sequencing comprehensively in the idea that common diseases might be a large and heterogeneous mixture of highly penetrant Mendelian subtypes. And the reality after the last 15 years is that it's now quite clear that it's a mixture of both. And as you heard from Matt, clearly even in less heritable and more extreme cases of intellectual disability, you can see a mixture of these entities in different proportions. So for various reasons, the first types of studies, largely for technical capability reasons, the first things we embarked on were using genome-wide genotyping arrays. And these enabled two fundamentally important types of discoveries. One is the less expected but widespread role of submicroscopic copy number variations, deletions, duplications, and so forth. And these were really the first fruits of the genomic era with respect to identifying risk for autism, schizophrenia, and other complex neuropsychiatric and neurodevelopmental phenotypes. And then secondarily, the detailed evaluation of the role of common variation. So a test of this hypothesis as to whether common genetic variation would play any role whatsoever in disease. And we come later to further technical advances, but I drift a bit into the work of genome-wide association studies and a little bit of the background on this. Now, the concept of performing a genome-wide association study was mostly, obviously, came from the technical capabilities to do such a thing. The theoretical idea is a very simple one, obviously, and it has its roots in both population genetics, in which it had become clear as the human genome was being sequenced with diverse components from different ancestry individuals that human genetic variation tracked with what was well understood from anthropology, which was that humans came very recently in evolutionary times out of Africa, and the vast majority of human genetic variation, or what makes any two of our chromosomes different one from the other, is ancient variation that dates back to our common heritage in Africa. This further was, extended theoretically into work, of population genetics over many decades, which suggested that the vast majority of genetic differences, given that that's where they reside in these common ancient variants, the vast majority of genetic variation that predisposes one to common human phenotypic differences would also be explained in this way. And so absent strong selection, this is how most human traits should work according to evolution and population genetics. Now, this was not a new idea, even with respect to something like schizophrenia, and it's more than 50 years now that an extremely influential but overlooked treatise on the polygenic nature of schizophrenia was written by Irv Gottesman and James Shields, and what they described is exactly what now comes to be known and commonplace in the human genetics literature as the polygenic nature of many, many traits. But it's taken us, obviously, most of that 50 years to get to the technical capability to test the hypothesis, and the last handful of years to really synthesize the data that we've observed. But in fact, what Gottesman and Shields and what many other quantitative geneticists at the time were postulating has exactly come to pass with respect to human disease, much as we recognized it came to pass with respect to animal breeding and animal traits many decades ago, but somehow resisted the idea that human diseases might be in a similar fashion. So with that in mind, the genome-wide association study era, I think everyone recognizes, has been extremely successful at identifying markers of risk and protection from disease of a very, very modest nature, generally speaking. So not highly predictive markers and not highly penetrant markers, but common genetic variation that attenuates risk in many, many different ways. So here, we started on this path at a time, and there was a lot of early success in late onset diseases, cardiometabolic diseases, as well as autoimmune and inflammatory diseases that were somewhat earlier onset. And unfortunately, when we came to almost every disease of the brain, and especially with respect to neuropsychiatric diseases, despite being more heritable and as common as many of these other phenotypes, the early genome-wide association studies were not successful at identifying new risk factors for schizophrenia. And so we were still left with almost nothing concretely known about the genetic origins of schizophrenia, even 10, 11 years ago at this point in time. So we had to face the question then of why this genome-wide association study strategy seemed to work more readily. And of course, the answer had to some extent, reside in natural selection. That in many cases, if we were looking at late onset cardiometabolic disease, there's no plausible, necessarily, direction of selection going on with respect to the genes underlying that. But with respect to autism and schizophrenia, and specifically, and more generally, almost any psychiatric disease, there is a huge fertility hit to having these diagnoses. And this has been repeatedly shown in Danish registry studies, Swedish registry studies, Finnish registry studies. Any place that has a decent registry would see this. And they almost recapitulate the numbers precisely. And that is that males with a diagnosis of schizophrenia or autism will have, on average, 1/4 the number of offspring as males in the rest of the population. Females with those diagnoses would have, on average, half the number. And this is a huge effect of selection against variation that gives rise to these phenotypes, even at a relatively modest level. However, the mathematics of it is such that the weakest effects, the types of things that we routinely find in genome-wide association studies, are almost immune to this sort of selective pressure. They're attenuated slightly downward, but you can still very, very easily have modulating factors that are increasing or decreasing risk by 10%, rising to high frequency in the population. So this is where the Psychiatric Genomics Consortium came in that the early failure of those genome-wide association studies to identify novel variants was discouraging, but enough members of the research community recognized that we had to, in order to give this hypothesis a reasonable test, work together, and that it would be by pooling studies from across the world that we'd have a much better opportunity to find genes. And fortunately, the historical competitiveness of the field of genetics was weighted by the fact that if we were all individually finding 0 genes, then whatever sharing of credits and so forth had to be happening, any fraction of something larger than 0 was larger than 0. So I would be remiss if I didn't at this point pause and note that one of the most important contributions to these efforts, not only in schizophrenia, but especially in autism and ADHD, has come from the remarkable partnership between iPsych and the Serum Institute in Denmark. And the Lundbeck Foundation's support for the iPsych program, led by many of my great friends, hopefully some of whom are in the audience. Actually, I heard Anders already asking a question, which is great. Hey, Anders. Has really led the way in terms of demonstrating how a nationwide health service and a nationwide effort to collect DNA samples can create an innovative that allows us to advance genetic research on behalf of the entire world. And so great thanks to my colleagues here who I've been very privileged to work with over the last 10 years, and great thanks to the Lundbeck Foundation for their visionary support of this fantastic effort. And of course, the other half of the project was led from the State Serum Institute. And this was where the entire country of Denmark deserves great credit for, and thanks from the rest of the world for having the vision and the foresight to not only store blood spots, but to create a mechanism by which it's possible for those to be utilized in important medical research. And so my thanks to everyone in the audience who is Danish for their support of this resource and continuing to make it available for obviously very secure and only highly approved research. But with this resource, and led by David Hogard and a great friend who passed away several years ago, unfortunately, Mads Hollegard, who really developed the techniques by which Guthrie-Card blood spots could be utilized effectively in DNA typing and DNA sequencing. And these studies will have a huge impact on our understanding for years and years to come. So anyway, the PGC study continued, sorry about that, to evolve and emerge over the first part of the last decade, and actually over the second part of the last decade. There'll soon be an update to this that brings the resources even further. But from those early studies, each of which individually was finding really no evidence of any substantive findings for schizophrenia, the combination of tens and tens and now, research studies from around the world has led to a remarkable explosion in our understanding of the genetic underpinnings of schizophrenia. When you compare the results from 2014 to the newer study, which is on its way to being published and led by Mick and James at Cardiff and Stefan in Berlin, the results continue to become more informative. I'm not going to go into a lot of technical details on that. I think what we start to see now is more of an emergence of biological themes and biological processes from these data that we didn't see in the earlier study. And so one very sensible question is, why do you keep going and making this GWAS larger? Well, now we start to only now get to the point where we can begin to extract some legitimate biological clues from it. And these are still very vague and general clues, but it tells us that we are on a pathway that ultimately from which will emerge more sets of concrete genes that we can pinpoint some biology from. And we start to pinpoint that biology as we look deeper and deeper. The other reason to increase the sample size of these studies is that you can begin to refine the mapping of loci. Even though we discover an association 5 years ago, we now, with the increased sample size, refine those associations, and we begin to, in some cases, pinpoint individual genes. We can understand that many loci have more than one independent association, helping to further pinpoint those genes, and a variety of prioritization techniques, mostly coming down to whether or not, or most of the information coming down to whether or not the genetic associations pinpoint a coding variant or likely functional variant in a single gene, or pinpoint only one gene in the locus, gives us a handful of genes with good and strong evidence. And this is too animated for this tapping thing here. But we now start to get from these refined data the ability to fine map down to individual variants and individual genes, which is really the target of the whole activity. Simply collecting associations is neither that interesting, nor is it particularly useful in terms of advancing our biological knowledge. And some biology has emerged from this effort over the past 5-6 years. My colleague Steve McCarroll led a fantastic study to dissect the tallest of these GWAS hits squarely in the MHC, but not corresponding to any canonical HLA types, but actually corresponding to, very nicely demonstrated, the copy number and therefore dosage of complement 4A gene. And this brought forth a very old, this is back into the limelight, in which the complement 4 being overactive might very well lead to the observational the observations made over several decades of potentially excessive synaptic pruning in late adolescence and early adulthood, seen sometimes in schizophrenia patients. Another example that I like to highlight is one of the rare examples, it's on an earlier slide, a gene called SLC39A8, which is one of the most pleiotropically and interesting genes in the genome with respect to this one specific amino acid substitution and its clear-cut association to a wide range of diverse phenotypes. And among those that are most notably interesting is the relationship both with serum and manganese transport that is defective in this particular common amino acid substitution, but also the opposite effects on increasing the risk of schizophrenia and decreasing the risks of Parkinson's disease, given the manganese toxicity epidemiology, which resembles a Parkinson's-like phenotype. And so here we are interested in both the biology that might be very well able to be intervened, but also the fact that there's a potential direct public health intervention, because my colleague Ramnik Xavier has shown that with the mouse knock-in of this variant, phenotypes in the gut and soon in the brain are recapitulated and reversed with a high manganese diet, and the phenotypes of the knock-in variant are mimicked by a low manganese diet. And so this gives us some opportunities that I'll touch on right at the end of the talk. Moving forward, though, we recognize that the results of these genome-wide association studies are not terribly biologically informative in the vast majority of cases. We have a lot of effort, and many people are putting a lot of effort into developing methods for extracting biological clues from this, but realistically, it's very difficult, as you can see from this picture, to extract concrete proof or evidence that any one of these genes and any mechanism influenced by any one of these variants has to be on the causal pathway to schizophrenia. So where does that leave us? Well, what genome-wide association studies thus far have done is give us several hundred points on the genome in which a common genetic variant is attenuating the risk of schizophrenia upward or downward. Several of these, but a very, very small number, give us some biological insight. But at the same time, this is only a part of the spectrum. And in fact, we might do better to focus on some other parts of the spectrum if our goal is to get to biology rather than to simply collect stamps and explain proportions of heritability, which of course it is. And so if we take where we were about 5 years ago with respect to this activity, we've established a role for common genetic variation. We've established that it's not so easy to extract biology from that. And obviously, during that period of time and the decade before, the appreciation, and going back a lot further with respect to 22q11, the role of large and often de novo copy number variants has been recognized as introducing not high penetrance, but higher penetrance and higher risk of development of schizophrenia over the lifespan. Now, there's several things that are missing from that graph, and that was really the focus of sort of a shifting or sort of moving then in parallel to a significant effort to approach exome sequencing and what it might teach us. And so this is something that we embarked on with, led locally in Boston by TJ Singh, and really a cast of thousands, all of the collaborators from around the world in the PGC and many other places that were beginning to embark on exome sequencing. And so the exome sequencing efforts have taken off slowly from the early days of exome sequencing, and there were a number of studies, as there were in the early days of autism and many other phenotypes, demonstrating that there is in fact a genome-wide excess of rare and disruptive gene-disrupting mutations, loss-of-function mutations generally, but also to some extent predicted damaging mutations. Very few genes were concretely identified from these, as it was challenging, as it was in autism, to pinpoint sufficient significant evidence for it to say that a single gene was responsible or associated to schizophrenia or autism, while it was comparatively straightforward to say that over the entire genome, there was a huge burden of such mutations, as Matt showed in intellectual disabilities. But fortunately, the exome sequencing accelerated over the next few years, and we brought together a consortium in the same model of the PGC to begin to pull these data together across many, many diverse studies. And almost all of TJ's work for several years was based on how to technically harmonize and working alongside the NOMAD team at the Broad Institute, which worked together to develop a lot of the techniques that we use, but how to harmonize when diverse sequencing strategies in diverse populations and just about every other source of diversity you can imagine was included in this effort. But fortunately, TJ was very persistent and also very skilled, and so was able to get through all of the technical aspects of that and move us forward into then genetic analysis ultimately, which in this case is really quite simple for the most part, and it's not too dissimilar from what Matt described, with the main difference being that we're not able to benefit from de novo mutations so much in the case of schizophrenia, because by and large we have cases and controls rather than complete families. And so, as with the work in autism and intellectual disability, we can see that, in fact, there is a substantial excess in cases of schizophrenia versus matched controls across many populations with respect to loss of function variants, and also with respect to those missense variants which are predicted to be damaging or which are clearly show evidence of being selected against, though as with the work that we've seen in the other disorders, the bulk of the statistical evidence comes from those loss of function variants, because they simply are much more precisely annotated as loss of function than we can at all separate deleterious missense variants from other missense variants based simply on the sequence context. But having done this, we can now, and this is work that's also soon going to be published alongside the updated PGC work, identify definitive statistical evidence for now about 10 genes, another 25, getting near that threshold, most of which are likely to be also significantly associated ultimately when the study expands. And from these, what's nice about it is we start to fill in some of the graph, and you can see, okay, these are tracking in a relatively similar way, not quite as common for the effect size as the recurrent copy number variants, but that's very likely because of the higher recurrent mutation rate of genomic disorders. And we start to gain some insights into biology because these types of mutations, as you can well appreciate, give us direction of effect in specific gene in a way that's much, much harder to extract from the common variant studies. And so as a result, we can now, generate hard evidence both from the ultra rare coding variants as well as the common non-coding variants that GRIN2A, not a surprising gene, emerges as a very, very strong candidate for schizophrenia. And what's really nice about this is this follows on a lot of literature and hypothesizing about glutamate receptors and of particular to us, since we also are very actively studying the genetics of autism. You'll hear about these efforts from Joe Buxbaum shortly, is that we see a great sort of parallel association where a very strong role for GRIN2B in autism is essentially replaced almost synchronously in some way with a very strong role for GRIN2A in schizophrenia, somewhat consistent with the timing of when GRIN2B and GRIN2A switch places as the dominant form of NMDA receptor construction. So just zooming a little bit further, I think basically what we see is, again, as Matt alluded to with respect to the DDID situation, not only do we see significant overlap in some cases between schizophrenia, DDID, and autism, but we also find very interesting instances where the types of mutations are quite different and distinct. For example, in this particular instance, loss of function mutations are conferring a strong risk to schizophrenia as well as sort of most forms of autism, and the intriguingly more severe outcomes of severe DDID are dominated by missense mutations that are predicted to be deleterious and clustered in certain domains of the gene, as Matt was describing. there's a well-established role in all of the phenotypes that you're going to hear described today for methyltransferases and demethylases, and I won't spend any more time talking about that. We're just about out of time. But what's interesting is that these are so consistent across so many phenotypes, and not all of them have a consistent presentation in every phenotype, but there's, the vast majority of these genes are hit in one or more of these three studies now that both autism, schizophrenia, and the DDID study that you heard from Matt are reaching a sufficient scale to really assess this question. And finally, I just want to I'll got a few more slides, but I think I'll rest on these points and really suggest that I think the next stages that we have to go through, and we've really been starting to develop cohorts here in Finland and elsewhere that will take us to the next steps of being able to now not collect a deeply phenotyped national cohort for the purposes only of discovery, but really for the purposes of now understanding much better what the discoveries, be they tens of genes coming from the rare variant side or hundreds of genetic associations and their combined burden. Now, pulling those data together and evaluating those in new cohorts that have lifelong medical data, that have a detailed patient assessment and medical and medication history, will allow us to then take the next steps towards better understanding of where we are going in schizophrenia. So thanks for your attention and the invitation to speak, and I'd be very happy to take any questions.
Many thanks, Mark, for this also very clear talk, and for also bringing the historical perspective, and thanks also for your opinion about where this type of research is heading to. We have a few questions. The first question from Anders Björklund. So thanks for an excellent talk, Mark. Could you comment on the sharing of both common and rare deleterious variation across neurodevelopmental disorders, for instance, between schizophrenia, autism, ADHD, and DD phenotype as defined in Matt? He's asking, I guess, DD phenotype as defined in Matt's studies.
Yeah, I'd be happy to. I think what we see with all of these phenotypes is that there's an extensive amount of sharing. I think we started to make these observations with respect to the copy number variants, which were extremely pleiotropic in their risk. In nearly everyone that was discovered in autism also conferred some sort of risk to either schizophrenia or DDID, or both, in fact. And in fact, the overwhelming signal that you saw in Matt's talk of rare and de novo mutations dwarfs what we see in schizophrenia and autism. I mean, by a long margin. And so in some ways, there's a tail of all neuropsychiatric phenotypes that share some of the risk factors that were defined in Matt's talk, but it's a small component of those, because the neuropsychiatric phenotypes that we're looking at are, of course, more, in some cases, much more common, and also far more heritable. And so these types of mutations are playing only one component. And I think what we start to see is that, in fact, the combination of risk is really what sets individuals on one trajectory or another, as it were. And the common genetic variation is equally, seen to be somewhat partially shared among most of the phenotypes that have been discussed here. I think it's more nuanced in the sharing in a certain way, because schizophrenia certainly shares risk with autism, shares a lot of risk with the other adult psychiatric disorders. But autism has a very interesting and unique overlap in terms of the common heritable space, because there what we see is quite conclusively that the inherited risk to autism actually corresponds to higher intellectual ability and higher educational attainment rather than lower, while of course the rare and severe mutations that are shared are highly deleterious to IQ and anything in that space. So we've got a lot to learn, but I think it's one of the more interesting ways in which we can begin to partition not only the risk to individuals, but also partition the biology, in that the shared components between two diseases are much more likely to describe coherent biological processes.
Thank you. We have a question from Vanessa Hall, and I think Vanessa had a comparable question to Matt before. What do you see in the future is required for further refinement of cohorts for GWAS exome studies, since this appears to be a key criteria for identifying novel variants?
So I think as I was saying right at the end, I think we do need to turn our attention to, having a more consistent and more a deeper phenotypic data on individuals, and these can come from national and lifelong medical systems, because now we need to focus not only on continuing the discovery process, and I think that's now becoming much more routine, but the ways in which we need to understand how each genetic variant, what forms of disease it's conferring risk to, where it is shared between diseases and where it's uniquely conferring risk to one disease or another, become much more important to start to dissect in terms of helping complement the biological dissection of the phenotypes that we study. And so I think we now have gone quite a long time out of collecting and putting together samples of convenience for the discovery power, and that's still obviously necessary. But now that we begin to reach that power, I think we also need to pay more attention to how we interpret the results coming from these studies.
We have another question from Anders. With the recent reports on STR expansions as a new class of rare risk variants in ASD, also non-syndromic cases, would you expect a role for this class of variants in schizophrenia and other neurodevelopmental psychiatric disorders?
Well, I expect a role for them in almost everything, because it's a quite a substantial category of genetic variation that we've to some extent overlooked. Now, I would say we haven't completely overlooked it, because our classical work, I mean, I find it kind of entertaining. This is described as a new source of variation, when this was really what some of us who are my age cut our teeth on back in the 1990s. It was all SSLPs and so forth as genetic markers for performing linkage studies and so forth. And from that time, I think we have a good recognition that actually there's a vast amount of simple sequence polymorphisms. Quite many of them, however, are not hypermutable, but simply faster, but still relatively slowly mutating, such that they have extensive linkage disequilibrium with neighboring SNPs. And so we probably, in some cases, find some of these, but these might be, in fact, highly enriched for these regions that are particularly complex to in terms of fine mapping, because if you have a length polymorphism, it may have certain alleles with certain LD to certain SNPs in the region. However, you're going to miss the association with any one or two or probably even three SNPs explaining it. And so I think this is a really interesting and area that we need to focus on, because it's probably the largest pool of genetic variation that's relatively untouched by all of the genomic technology of the last decade.
But real quick, a last question from Konstantin Kodosevich. Great talk. For missense mutations, these could change the function of the coded protein. Could this information be integrated in prediction of disease mechanism?
Yeah, absolutely. I think there's a lot of work. I've even had a few postdocs working in this area the last couple of years, trying to do a better job of predicting, for example, which missense variants in specific gene families, such as sodium channels and so forth, are likely gain of function and loss of function, and trying to use that information to better develop genetic association models to more accurately characterize what's happening in a particular gene, both on an individual patient by patient level, but also to improve the gene discovery models that we apply. And so I think that is something that we need to begin to pay more and more attention to.
Very good. Thanks very much again, Mark, for your great talk, and the audience for the nice questions. And now we move to the flash talks. So there will be 3 flash talks of 5 minutes. The first one is by Daniel Kalame from Baylor. He will talk about biallelic loss of function variants in the splicing regulator and NSRP1, causing a severe neurodevelopmental disorder with spastic cerebral palsy and epilepsy. Daniel.
All right, thank you very much. And thank you to the organizers for allowing me to speak today. So again, my name is Daniel Callum. I'm a child neurology resident and postdoctoral fellow in Jim Lepsky's lab here at Baylor College of Medicine. And again, it's my honor to present our work entitled Loss of the Splicing Regulator NSRP1 Causes a Severe Neurodevelopmental Disorder. Let's see. So alternative splicing, of course, is a major contributor to transcriptome diversity. Splicing patterns are shaped by RNA binding proteins known as splicing factors. These splicing factors bind to specific pre-mRNA sequences, and through their interactions with the spliceosome, selectively promote exon skipping or inclusion, use of alternative 5' or 3' splice sites, or even intron retention. Therefore, the specific combination of splicing factors expressed within a tissue or cell determines its transcriptome. Mouse genetic studies have elegantly illustrated that splicing factors play a critical role in mouse brain development by regulating neurogenesis, cortical lamination, Purkinje cell migration, synaptogenesis, and synaptic homeostasis. Despite this, few human neurodevelopmental disorders are known to result from splicing factor dysfunction. Nuclear speckle splicing regulator protein 1, NSRP1, is a ubiquitously expressed splicing regulator not known to underlie a human neurodevelopmental disorder. Found within the nuclear speckle, a major hub for splicing factors, NSRP1 interacts with and counteracts the splicing activity of the arginine serine-rich splicing factors SRSF1 and SRSF2, thereby promoting either exon inclusion or exclusion in a gene-specific fashion. Unfortunately, mouse knockout of NSRP1 results in early embryonic lethality, which has limited our understanding of its role in mammalian neurodevelopment. Through exome sequencing rare variants, family-based genomics, and the online matchmaker gene matcher, we have identified 3 consanguineous families with a severe recessive neurodevelopmental disorder, which segregates with homozygous, ultra-rare loss of function variants in NSRP1 as seen here, with a total of 6 affected individuals. In it, loss of NSRP1 in man appears to cause a very severe neurodevelopmental disorder, with common clinical features including significant developmental delay, epilepsy, axial hypotonia, and appendicular spasticity, as well as microcephaly, dysphagia, and mild dysmorphic facies in most of the patients. And you can see examples of the patients to the right there. Loss of NSRP1 also causes brain abnormalities. On review, we noticed that there was a consistent pattern of a simplified gyral pattern, as well as underopercularization, which is illustrated by the red arrows, and some degree of cerebellar vermian hypoplasia, illustrated here in this figure by the white and black arrowheads. NSRP1 is located on chromosome 17, and it consists of 7 exons. Its protein product has 2 RNA recognition motifs, an arginine serine-like domain, 2 overlapping coil-coil domains, and then a critical nuclear localization signal near the C terminus. The nuclear localization signal has been previously shown to be required both for nuclear localization, but also for the splicing activity of NSRP1. The variant that was found in family 3 is a nonsense variant in exon 2, and therefore should result in the severely truncated protein or be subject to nonsense-mediated decay. Whereas the 2 variants in families 1 and 2 are frameshift variants in the last exon that should escape nonsense-mediated decay, but cause loss, or they're predicted to cause loss of that critical nuclear localization signal. In conclusion, biallelic loss of NSRP1 in man appears to underlie a severe neurodevelopmental disorder characterized by spastic quadriparesis, epilepsy, and microcephaly, and further study of NSRP1 via patient-derived iPS cells and neuron-specific knockout mice will provide insight into its role and the downstream gene conformers involved in CNS development. I'd like to conclude by again thanking the organizers for allowing me to present here, as well as my mentor, Dr. Lubsky, all members of the Lubsky Lab and affiliated labs, the program leadership of our neurology residency program, including Gary Clark, Timothy Lotzi, and Tuan Chow, and all of our collaborators and our funders. Thank you very much.
Thank you, Daniel. So we'll take questions at the end of the 3 talks. The next speaker is Stamatina Tsanoulinou from the University of Lausanne in Switzerland, and she will tell us about the inhibition of TRPV4 rescues circuit and social deficits unmasked by acute inflammatory response in a SHANK3 mouse model of autism. Stamatina.
Yes, thank you very much for the introduction, and thank you for giving me the opportunity to present to you our latest work regarding SHANK3 mouse models of autism. So first of all, I'm going to talk about briefly what is SHANK3 and why we study it. So SHANK3 is located postsynaptically and is a scaffolding protein at the excitatory synapses interconnecting glutamatergic receptors. And in fact, among the synaptic genes that are implicated in autism, SHANK3 mutations are one of its most common monogenic causes. And moreover, what it's known is that haploinsufficiency of SHANK3 has been linked with Phelan-McDermid syndrome that actually presents with often severe intellectual disability and also severe autistic-like symptomatology. However, although there are many SHANK3 knockout models, mouse models, heterozygote mice, few heterozygote mice are showing a severe phenotype or actually social behavior problems. And in fact, this is an important point because heterozygote mice are considered to be closer to the human condition. So in fact, we wondered whether an environmental challenge or in this case an immune system activation challenge in our case would lead to unmask a vulnerability in the social domain in heterozygote mice. So for this purpose, we had a model of knockout of SHANK3. There are many models as we discussed. And we had wild-type littermates and heterozygote mice where they were challenged with LPS injection or a lipopolysaccharide that is a potent activator of the immune system 24 hours before we assess their behavior in the 3-chamber interaction assay. So as you can see on the left panel, the heterozygote mice with LPS, despite the LPS injection, they showed an intact sociability, preferring a social over an object target. Heterozygote mice with vehicle, they showed also an intact sociability as we expected from the literature. However, the heterozygote mice with LPS injection, now we could see a deficit in the social behavior. So our question was now which brain areas or cell types could be involved in what we were seeing. So we focused on the NAC of the nucleus accumbens for its involvement in social behavior. And in fact, we downregulated SHANK3 locally in the NAC with a viral approach. And we saw that these mice had both reduced sociability and also D1 medium spiny neurons hyperexcitability of the NAC. So in order to understand better the mechanism behind this, in collaboration with the laboratory of Professor Denise Abaudon, we performed a FAC sorting and RNA sequencing experiment to understand what the genes that are up or down regulated. Among the genes that were highly upregulated in D1 medium spiny neurons, we saw there was a channel TRPV4 or transient receptor potential vanilloid 4 that was, of course, caught our attention because it was highly upregulated, but also because apart from being involved in many cell processes, it's known to be involved in calcium influx, and thus it's considered to be an important regulator of neuronal excitability. So to understand whether it's relevant for what we are studying, this TRPV4 change, we went back to our heterozygote mice, and now we patched D1 MSNs, D1 medium spiny neurons in the neck after a challenge with LPS. And we can see, as you can see on the orange line, the number of action potentials is higher in these cells, meaning that we are mimicking what we saw with the downregulation of SHANK3 in the neck. However, when we bath apply an inhibitor of TRPV4, we see that the levels of excitability are back to control. But what does it mean for in vivo? So we went for in our heterozygote mice again, and now they were cannulated above the nucleus accumbens. And before doing the experiment, of course, they were challenged with LPS 24 hours before. And now the mice were infused in the neck locally, either with vehicle or with TRPV4 inhibitor. And we could, in fact, see that with vehicle injection, we could recapitulate or confirm what we saw before, unmasking the vulnerability in these mice. But with the TRPV4 inhibitor, we could see an amelioration of the ASD-like symptoms. So I showed you in sum that immune system activation by LPS could reveal social behavior deficits in SHANK3 heterozygote mice, and that TRPV4 inhibition of TRPV4 could rescue circuit and behavioral alteration in these mice, specifically by focusing on the nucleus accumbens. So I would like to finish by thanking, of course, my lab and our collaborators, our funding sources, and you for your attention.
Thank you, Martina. We move to the third flash talk by Andrea Asenjo Martinez from the Biotech Research and Innovation Center at the University of Copenhagen, Denmark. And this is about the identification of diseased neuronal cell types in 15q13.3 schizophrenia mouse model. Andrea.
So thank you for the introduction, and I would also like to thank the organizers for giving me the opportunity to present part of my PhD project today, which aims to identify diseased neuronal subtypes in schizophrenia. So in the brain cortex, neural networks are formed by cortical projection neurons that are excitatory and interneurons that are inhibitory. And it is really important that excitatory and inhibitory signals are well balanced in order to have proper brain activities. And both families of neurons are really diverse, especially interneurons, that we can see they have many different morphologies, they have different electrophysiological properties, and also the different gene expression patterns, with more than 60 transcriptomic subtypes divided in 5 main families. So when interneurons are not working properly, this balance between excitation and inhibition is disrupted, and that leads to the appearance of neurodevelopmental disorders such as schizophrenia. And we hypothesize that different subtypes of interneurons are affected differently in schizophrenia. And to assess that, we use single-cell RNA sequencing technology. So for this study, we were using one of the most robust schizophrenia mouse models, that is the 15q13.3 microdeletion, which is based on the rare recurrent copy number variation found in patients. And it consists of the deletions of 7 genes in the chromosome 15. So we performed single-cell RNA sequencing in control and schizophrenia mouse brains, and we could identify and annotate 31 different subtypes of interneurons divided in those 5 main families. So first, we integrated both control and schizophrenia dataset, and overall we can see that the alignment was pretty good between them. However, for example, in those small clusters in the center of the T-cine plot, we could see that there was a visible transcriptomic shift between control and schizophrenia datasets. And then we looked at the compositional analysis of the different subtypes, and we could see that in some of them, there was a reduction in the number of neurons in schizophrenia versus control. So then, in order to identify disease transcriptomic affected subtypes, we use gene expression similarity score that is based on the Pearson's correlation between and within conditions. So a similarity score bigger than 1 means that that subtype has more transcriptional changes. And here on the plot on the right, you can see all the different subtypes ranked by the similarity score. And this means that the ones that are on the right side of the plot are more affected, and their transcriptome is more different between wild type and schizophrenia, which might indicate a schizophrenia-specific effect on their transcriptome. So moreover, we did a hierarchical cluster analysis where we separated on each subtype control and schizophrenia cells. So we see that in some subtypes, both conditions were clustering together. In others, they were not clustering together, but they were coming from the same branch. And in other subtypes, they were not even coming from the same parental branch in the tree, which indicates the transcriptome are more different. And this was matching with the data that I showed you before about the genomic correlation. So we took in consideration all these different types of analysis for selecting subtypes that appear to be more affected for further analysis. So first, we validated the compositional analysis. We validated the compositional analysis of single-cell data. For example, here you can see that we validated the subtype EB8 that showed a reduced number of neurons in the single-cell data. And here by immunohistochemistry, we were also able to see this. And it's important to mention that we could identify different markers that specifically could label the specific subtype that we were validating. So then we continue with the gene ontology analysis of these subtypes, and we could see that there were many interesting pathways that pop up in this analysis, and most of them were related in this subtype with synapse assembly and synapse membrane. And also when we were looking at the differentially expressed genes, we also saw genes that were differentially expressed specifically only in these subtypes, and those genes were related as well with the geo pathways that we found in this subtype. So to conclude, I hope I could convince you today that not all the subtypes of interneurons are affected equally in schizophrenia. We could see by gene correlation and hierarchical cluster analysis that the gene expression pattern is more disrupted in some subtypes than others. Also, some subtypes show a reduced number of cells by single-cell RNA sequencing, and we could validate this reduction by immunohistochemistry. And also that there are subtype-specific geoterms, and this suggests that there are different contributions of the subtypes to the phenotype of schizophrenia. And with this, I would like to thank all my lab for their support, and thank you all for your attention.
Very good, thanks very much. We have one question here from Prakash Devaraju for Stamatina. He could not get the connection between the TRPV and LPS. How is an ion channel, how is modulating an ion channel ameliorating LPS-induced effects?
Yes, so this is a very good question, and it goes to the mechanism, to the actual mechanism of how, with the deregulation of SHANK3, we have an upregulation of TRPV4, and how we see that in heterozygote mice by doing this immune system activation. We don't know the molecular cascade. It could be mechanical. For example, it could be that this is a scaffolding protein, SHANK3, so when you downregulate it, you are affecting the connection, and then as a compensatory mechanism, you have an induction or an increase in TRPV4. So this is something that we definitely would like to explore in the future, but we don't have an answer of the actual mechanism, molecular mechanism. But they are connected, and in fact, we also saw that after the LPS, we tested the interleukin-1 beta, TNF-alpha, and TRPV4. And in fact, we saw that TRPV4 was increased only in heterozygote mice and not in wild-type litter mates. And also to mention that TRPV4 by itself is been linked with immune system activation. So it's already known that it's participating in inflammatory processes. So the link is not far away, but we just need to find the details of the mechanism.
Yes, thank you. I would have a question for Andrea, actually. Could you perhaps speculate on the reason why there is differences in the transcriptomic effects across neuronal cell types?
Why? That's your question?
Yes. Yeah.
Okay, because yeah, we think the interneurons are affected different, and also they can be affected at different time points during development. And yeah.
Okay, I don't know if you could, have you considered perhaps a different genetic profile of these different cell types that may vary, that may make them more susceptible to developmental alterations that would ultimately be visible through the transcriptomic differences?
So, of course, now in my PhD project, we study the transcriptome, but of course, epigenetics also play a role in this. And also, it's really important to know like the different connections and electrophysiological properties that these neurons could have, going back to your first question. So, depending on their connectivities in the neural networks, that's why they can be more or less affected in schizophrenia.
Okay. We have a question from Marianne. They talk to find difference between male and female in connection with inhibition of TRPV4. So I guess that's for Stamatina.
Yeah, so unfortunately we didn't look at females, but really we are making it part of our studies in the future to understand, because so one reason is because males are more affected in autism models, but it's not an excuse. So we should look at females. I'm really in favor of changing the trait. So it's actually part of our future projects, but we don't have an answer for male-female differences.
Last question from Emna Shebi. I assume that's for Andrea. So thank you so much for the informative presentations. I'm curious to know how you found the contribution of genetics in having better insight on the function of different neurons, as well as the study of the circuits.
So, as I said, we could identify different markers that were specific for each subtype. So also the different subtypes are located in different layer, the layer of the cortex. So taking into account the morphology, the connectivity, and the markers, we are able to trace the different functional data to our transcriptomic subtypes.
Good. Last question for Daniel. Have you... sorry, the question disappeared suddenly. No. It was voted for, so it went up the rank. Have you looked at NSRP1 heterozygous mice? If your patients are heterozygous? First question. Second, are you sure that the mutation in family 1 leads to NMD? It could be rescued by a downstream start codon, for instance, ATRX or R37X.
Right. So we haven't looked at, I haven't personally looked at heterozygous knockout mice. The knockout mice were generated some years ago by a Korean group, and reportedly they were healthy. I think the only phenotype was they were a little smaller than wild type mice, but it would be interesting. And then for the second part of the question, that certainly is possible. We think the segregation data in the family is pretty strong evidence for the pathogenicity of the variant, but certainly it is possible that that variant may not be subject to nonsense-mediated decay.
Okay, very good. So thanks again, Daniel, Stamatina, and Andrea for the interesting session, and thanks to the audience for the questions. Now we move to the next talk by Joe Buxbaum from Icahn School of Medicine at Mount Sinai. And Joe is going to tell us about the gene discovery to novel therapeutics in autism spectrum disorder.
I would much rather be there in person. But one of the first things I'm going to ask everybody to do is take out your book and change the title of my talk, because I need to lower expectations. So The original talk was a highfalutin from gene discovery to novel therapeutics, etc. And I'd like to change the talk title to Carrying Coals to Newcastle, because everything I'm going to say you've already heard about from the experts in the field, or you will hear about. So, how do I advance slides? Right. So this is Newcastle, and these are coals leaving Newcastle, and I'm sure you're familiar with the phrase coals to Newcastle, which reflects the idea that sometimes some people like me try to bring something to the top site in the world. What's interesting about this phrase, and there's many phrases across multiple countries, is that it was actually first came out in 1583, first recorded in 1583 by James Melville. And he was trying to reinterpret an historic Greek phrase, which is owls to Athens. So this is his text, Coals to Newcastle. And James Melville, oops, wrong way. James Melville, if you look on the left, you'll see, if you're a trainee, you see had multiple last names, multiple spellings. And so if you are new to science, you want to make sure that you choose a single name and a single initial that you use consistently so you can find yourself in PubMed. If you don't have a middle initial, by all means, go get one. I recommend the initial X. So then it'll be JX Melville, spelled one of three ways. But James Melville was quite famous in his time, a reformer, and his uncle, Andrew Melville, is actually the person that came back from training across Europe, including France, and actually revitalized all the major universities in Scotland, and was directly responsible for forming the University of Edinburgh, which, as you know, one of our awardees has been at for many years. And the phrase coals to Newcastle was first written down exactly the same year that the university was founded. I took the liberty of taking Adrian Bird's photo online and flipping it so that they'd both be looking the same way. So now you know why I feel like I'm bringing coals to Newcastle. Well, I will explain why I'm bringing coals to Newcastle, or owls to Athens. And the reason is because I could tell you this radical new idea that if we have a new disorder and we describe it clinically, and we find the cause of the disease, and we make model systems, we have family registers, do natural history studies, and do in vitro preclinical work to reverse the phenotypes, that will lead to clinical trials. But of course, this is something of a deep fake, because this is actually from this paper over here, which talks about 50 years of Rett syndrome. And it goes without saying that Rett syndrome is the paradigmatic example of this sort of approach, with, of course, some of the most important seminal discoveries in this timeline made by our two honorees: identifying the genetic cause of Rett syndrome by Huda Zoghbi, the first animal model by Adrian Bird. If you go to the right, the reversal of the Rett phenotype in the mouse model was first demonstrated by Adrian Bird. And the net result of all of this, of course, is that there are now multiple clinical trials in Rett syndrome based on this etiological discovery, the formation of these large registers, and a lot of preclinical work. So I don't really have much to add to this story in some ways, although I thought for a moment I could talk a little bit about different kinds of approaches. For one of the things that you might hear from Huda is that, when she was at Texas Children's, she saw a couple of cases of Rett syndrome, and consistently and persistently began to collect more and more families. She wrote a paper, I think it was 95, and then families began to come to her, and she had a lot of individuals with overlapping phenotypes. 1985. That then led to the discovery of the gene. So that really is that is the classic gene discovery approach for clinical genetics. But I'm going to talk about, for me, I need to know how to find genes if I'm lazy and I'm not going to get many people with similar phenotypes. The other part of the issue you've already heard about from Matt Hurls, and again, coals to Newcastle. There are many disorders that have an array of overlapping symptoms. So these are the 7 that I'm kind of putting together here: structural brain abnormalities, congenital malformations. You saw Matt talked about heart abnormalities, for example, motor delay, language delay, LD or ID, epilepsy, EEG abnormalities, and some kind of severe psychiatric disorder, which you could actually put in schizophrenia or even atypical schizophrenia and atypical bipolar. So it's much harder here, excuse me, much harder here to say we're going to collect 10 families with exactly the same syndrome, because these are what I'm calling non-specific syndromes. And they come in every flavor. Excuse me, this is an actual fully complete Venn diagram for 7 overlapping syndromes or anything. And in any case, any individual person can have all 7 of these, 2 of these, any 3 of these, and so on. If you want to find this Venn diagram, if Matt Hurls and the DDD want to put all of their patients into one of these colored boxes, it's from the Electronic Journal of Combinatorics. So Matt's told you in significant detail how do we go after these non-specific syndromes, which is something that we can only do now because of the availability of very cheap sequencing. And then on the other extreme, of course, there are ultra rare syndromes where you're not going to be able to bring together 10 or 15 people with the syndrome. This is the really groundbreaking work of Timothy Yu for an N of 1 treatment that happened over the course of 18 months. And again, as you've heard both from Matt and from Mark Daly, we now know enough about the genome and what to expect that we can actually make a reasonable prediction about an N of 1 syndrome, especially if it is a de novo mutation. So this is an example of that work that Matt Hurls just talked about. This is one of the DDD papers, one of the first ones, showing an accretion of mutations in multiple genes across the DDD, and then showing genes that were recurrent. And the one on the right, DDX3X, I'll come back to in a little bit. So what I'm going to do, since I've really have little to add to what you've heard before and what you're going to hear from Huda Zoghbi, is I'll take a little bit of a look back and a look forward about the Autism Sequencing Consortium. I want to just say that this logo was the only time in my entire career I actually made something graphic, and I want everybody to use it. The problem is, as you can see, it's in the old format of slides. It doesn't fit on the 16:9, so I don't know what to do. But there are multiple leads in the ASC, including Mark Daly, who you just heard from. And the ASC started, these 2 individuals started the ASC. One of them is retired after that, and the other one just came back from retirement. And this is the 2 of them signing an act in 2009 called the the ARA Act. I can't remember all the full acronym. And some of that money, through intervention of Congress, a tiny bit of the ARA support went to the NIH and provided the basis for some novel ideas. And so around that time, particularly several people, particularly me, strong-armed a bunch of people to come to New York City and to talk about this new idea called the Autism Sequencing Consortium. And I want to answer 2 questions right away. Why is everybody so dour? And where is Mark Daly? So dour, you can see I'm smiling rather happily, and everybody else is looking kind of grumpy. But where is Mark? So Mark was there. Mark was there, but if you know Mark, he had to leave the moment the meeting ended. In fact, we ended the meeting on his schedule, and he was on a plane or maybe a train. So he left immediately, did not come to the wonderful and critically important launch photo. And the reason people are so dour is because we basically said what we'd like to do is that people share data before publication, that if you wanted to be in this consortium, you had to share your data before you published it, either sequence data or DNA. And that was a pretty radical idea in the US at the time. Obviously, you've heard from Matt on a national level, and you've also heard about iPsych. People are very happy to do this. But here, everyone in the room had received a small amount of money to do a small study. And we were saying, we'd like you to share that data and not publish it first. And they're dour because Thomas Lenner, who's the person on the left, and Alan Packer and other funding agencies were in the room too. And they said, we think this is a good idea. So there was going to be some pressure brought to bear on this concept of prospective data sharing. And in the midst of all of this, funded by the ARA Act, several groups began to do some exome sequencing in autism. This paper over here, which came out in 2012, where Ben Neal was the lead author and Mark Daly was the senior author, 5 different sites coordinated the sequencing of about 200 autism trios. There are 175 in the final paper that passed QC. So it took 5 sites, including 2 big genetic sites who knew what they were doing, and 3 sites that were not genetic sites or not NIH genome sites, including Mount Sinai, who very much did not know what we were doing, to sequence 175 trios. And the big finding were there were a couple of genes, CH8 is mentioned on the bottom left, but the big finding was that the rate of nonsense de novo mutations was elevated, as shown in the box, compared to expectation, which was derived by various models at that time. And so if you see below that, the p-value for that difference was p.04, but please note this was one-sided. So you know what it means when you have a one-sided p-value. It means it wasn't significant when you did a two-sided p-value. But we knew we were on the right track, and we were not the only people doing it. There were multiple excellent groups, Mark Wigler, Matt State, Evan Eichler, looking at data from the Assignment Simplex collection. So in 2012, there were 4 papers that came out for a total of 1,000 ASD families looking at de novo mutations, particularly a loss of function single nucleotide variation, and collectively identified 9 genes with significant evidence for linkage in autism. And so the ASC, the Autism Sequencing Consortium, put this to the NIH and basically said, as you said, that we will only find autism genes, these that are impacted by a rare deleterious variation, if we sequence on scale. And this was some kind of analysis of gene discovery as a function of number of cases under different models of the number of autism genes. You've just heard Matt say that there are 2,000 genes that they use as part of the DDD. We estimated at the time that there were something around 1,000 autism genes. And so, as you can see, with 1,000 autism genes, we need many, many probands in these trios to reliably find those genes. So that was the basis, the rationale for the ASC. It was funded. And what's made the ASC successful, and I think it is successful even though I'm directly involved, has been enormous philanthropic foundation and government support. This concept of data sharing and even prospective data sharing, which, as I told you in the beginning, was a bit controversial, shall we say, but also sharing across other data sets, the DDD, ISAC, UK10K, FinGen, and so on and so forth. The development of novel statistical tools. This has been critical for all the studies you've heard from Mark and Matt about, and we haven't gone into in sufficient detail, but many people have made it their life work to help us get as far as we are, including, of course, Mark and the folks in Matt Harrell's group. And then sequencing, the cost of sequencing going down, the efficiency, and our ability to do it very rapidly. So in 2014, we had our first paper where we actually found this time significant genes. I won't go into this. I will point to the right, one of the figures in the paper, showing this idea that even though these are all autism cases, the genes fall into many of these non-specific phenotypes, or phenotypes associated with what I called before non-specific syndromes. Okay, so ASC, the second major paper, or third, depending on how you count, many people were involved. These people took the lead on the secondary analysis. This paper came out in early 2020, a year ago. And very briefly, it had these, just like multiple genes were identified as autism genes. Each of the genes have multiple mutations in that gene, genes of major effect. Everything above, towards the top with the very low p-values are highly significant genes with, as you heard before from Matt and from Mark, highly penetrant in most cases, at least if they're towards the top. You've also heard that the genes broke down generally into the green genes, which were neuronal genes, neurocommunication genes, and genes that regulate other genes, or the purple genes, or the GERs. One of the interesting hypotheses that were floating around was that the primary impact of the GER mutations was to regulate somehow the expression of the neuronal genes. And in this analysis, and it's not the final word, but in this analysis, that proved not to be the case. So the green genes are the bottom circle on the circle on the bottom, and there's a p-value there. But all it really says is that if we look at all the GERs with known targets, they are not actually doing much to the neuronal genes. In other words, there are two ways to get autism: early development GER, and possibly later in development through synaptic or neurobiological changes. There's also two ways to get autism if you think broadly about the brain having excitatory and inhibitory neurons. This is going down the excitatory lineage over time, which is the bottom, the oblong on the bottom of each of these two panels, or across the inhibitory lineage, which is the weird shaped thing on the top. On the right-hand panel, you see the red. That means more expression. And what you can see is the genes that we're finding are expressed both in inhibitory and excitatory lineages. And so again, it's not going to be the case that a single subtype of nerve cell, for example, is going to be causing autism. This Matt showed you very elegantly. If you look at panel A, there's a plot of what kinds of mutations, what kinds of mutations are being found for each gene. If you look at ADMP, bottom right, it's all protein truncating variants. If you look at DEF1, which is on the left in the middle, it's entirely missense variation. And as Matt already showed you, that tells you something about what you're seeing. So missense only in a specific domain. These are probably going to be some kind of gain of function or dominant negative. And you've also heard some very elegant work on TRIO by Mark Daly. So as we accrete more mutations, we can begin to understand more about what is actually going on. Again, as we have more genes with mutations in them through ASC or DDD, we can start to look into copy number variation, some of the recurrent CNVs, and try and find the genes that contribute to significant parts of the risk. It's no question in my mind that some copy number variants that are recurrent are, in fact, contiguous gene syndromes, but some of them have a specific gene that accounts for most of the risk. SHANK3 is a good example that you just heard about. And we have, as more sequencing goes on, we can begin to dig into these recurrent copy number variants, these pink regions on the two panels F and G, and find genes that seem to be contributing most of the risk. On the right, you see SHANK2, which is a close friend of SHANK3. So here's an ASC study on the left, and here's one of the DDD papers on the right. And what I'm trying to make the point here is that some of the genes, many of the genes overlap. ADMPs in both, SNC2A, SYNGAP, FOXP1. We're finding overlapping genes. And that led to the question about whether they're all the same genes. And there's been a bit of a online discussion about that, which I'll come back to very briefly. But I wanted to look a little bit forward at ASC3, the next version of ASC, which includes more data from the ASC, but most importantly, includes data from the SPARC cohort, integrates all the data from iPsych, which you've already heard about on the sequencing side, Simon Simplex collection, but includes SPARC, which is a very important Simon's initiative, which has now 27,000 sequenced individuals. And I won't go into it except to say that the brilliant work of Stefan Sanders, Mike Tolkowski, Jack Fu, Carl Satterstrom, Harrison Brand, Shandong. I should also mention Ryan Collins, Bernie Devlin, Catherine Rorty, Mark Daly. That guy Bucksbaum didn't do much. There is now a much larger list of autism genes, 186 at a more stringent threshold than we used before. Very elegantly, this group integrated genomic copy number variation, so using the GCMV protocol, calling copy number variation from the exome sequencing, as Matt also showed you. And then now we can actually get very strong data, and this is, I won't belabor this, but this is also what Matt showed you. We can now take genes and map them, whether the primary evidence from a truncating variant, a missense variant, a CNV or otherwise. The second gene on this list is SHANK3. From the left, you see that the red is a PTV, and the purple is a CNV, a deletion. And one of the things that we're able to do together is collaborate with the DDD and put the DDD findings together with the ASC findings. And the combination of significant genes is 352 genes. And we do see some genes that are truly appear to be autism specific, more likely to be inherited, much less severe phenotypes, and so on and so forth. So I'm going to look forward a little bit in two ways. First is, I said we're going to, the original title was suggesting we'd find some genes and do some precision medicine. Given what we know about MECP2, we know that's an approach. So I'm going to ask a slightly different question. Is there really a viable alternative? And I would say there isn't at this point. There's not a single pathway. I went through that pretty quickly. What's positive, and Matt mentioned this, is that most of what we're finding, because we're powered to find these things, is dominant. In other words, there's a second allele that could be manipulated. They're primarily loss of function, and I showed you, and Matt showed you, and in fact, Mark showed you too how we can figure that out. So that means, if we have one null allele and then we have a healthy allele that we can increase its activity, its stability, you name it, we have a target there. These genes tend to be U-shaped in terms of their impact. You heard mentioned yesterday by Adrian that MECP2, when you overexpress it, is deleterious, and that was shown by Huda Zoghbi, who will no doubt talk about it. But we know this from many copy number variants, a deletion or a duplication have impact, and some of the genes where we actually have, for example, duplications like MECP2, or balanced translocations where you could have monosomy and trisomy in the child, we know that too much is also too bad. It's been called now, I called it U-shape, that's the old phrase, now it's called the Goldilocks effect. Tend to be severe. I don't think I need to explain that further. This is not autism, this is autism with intellectual disability, language delay, motor delay. And in multiple examples in preclinical work, if you turn the gene back on, things get better. And of course, the first person to show that was in MECP2 was Adrian Bird. They are, quote, fairly common. I don't mean they're common. I mean they're common enough for a biotech company to be interested in an orphan designation. And the top ones, at least, are highly penetrant. They account for much or most of the risk. So I don't see how we can get away from precision medicine at this point. So I'll give you 30 seconds about what we're doing in the Seaver Autism Center. We picked a few of those top genes, which are molecular targets. We try and make human neurons in vitro. We make genetically modified rodent models, and we try and see as many patients as possible with those mutations. The four that we work at, and we just started with a fifth one called CHAMP1, ADMP, FOXP1, and SHANK3 are the ones we worked on the most. SHANK3, don't need to talk about it. You just heard a great talk from Stamatina. It's what I usually talk about. It's a rather boring talk at this point. CHAMP1 is new, but I thought I'd talk about DDX3X. And DDX3X is particularly interesting for a variety of reasons. Firstly, it was shown by the DDD and colleagues that it is the major cause of unexplained intellectual disability and developmental delay in girls. 1 to 3% of ID and DD in girls is caused by mutations in DDX3X. It also is implicated in autism. This is the work of Dennis Wall and Dan Geschwin. And many people have data for the same gene in autism. This is a recent paper from the DDD showing that if you look at the observed overexpected in terms of mutations in DDX3X, all the way at the top there in females, massively increased rates of mutation DDX3X over the expectation, and even higher than MECP2 in this study. And I would say that it's probably the case that DDX3X is more common than Rett syndrome. So what we do, as I mentioned before, is we bring in individuals with this syndrome, and we do a multi-day. This is a 3-day deep phenotyping with biomarkers and so on. On the top left, you see the 15 patients we were able to bring in before COVID. Below it shows some of the recurrent ones in the literature. These are the pictures of some of the girls and one boy, by the way. Like Rett syndrome, boys with a serious mutation will not survive, so it's predominantly girls. What you see on the right, is our very extensive phenotyping, and not even talking about the biomarkers that we add as well. 80% meet criteria for ID, 60 for ASD, 50 for ADHD. This is the work of Paige Cypher and Dorothy Grice. They led the work. And I won't go through all of this, but there are sensory abnormalities, IQ abnormalities. What I'd like you to see on this slide is that we color the PTVs and the missense separately. And for some of the traits, like you look at the DQ or IQ surrogate, if you will, everybody on the top right that is higher, has higher RQs and even typical IQ, has a PTV. Well, anybody with a missense mutation has a lower IQ, a significantly lower IQ. Second panel on the right going down, less severe autism using a severity metric is going to be much more likely to be associated with somebody with a PTV, a protein truncating variant, rather than a missense. And there are multiple phenotypes that are like this. On the left, you see sensory abnormalities, and sensory issues are very profound in these cases, but that is not associated with the different kinds of mutations. This is just looking at the various things that we looked at. Adaptive behavior, some of the sensory behaviors, language skills, and so on, showing even in a tiny sample of just 15 because of COVID, there already are significant differences, and those with a PTV are less severe than those with a missense. Very quickly, the second arm of the study we do with DDX3X is being led by my colleague Sylvie Duroubaix and her team. This is now in bioRxiv. If you look to the bottom left, these are mouse models. You see there's a 0 there, and that is the absence of males born with carrying a mutation DDX3X. So just like Rett syndrome, you just don't find males with a deleterious mutation. You will find males with a hypomorphic mutation, also like Rett syndrome perhaps. And this is all online, but you can see that there are deficits in cortical lamination in these animals. Comparing the heterozygote to the homozygous wild type, you can see the CTIP2 positive cells are mislocalized. And I won't go into it, but Sylvia and her team have done a great job to show that there are motor delays, there are sensory delays, there are brain changes, changes in cortical lamination, there are behavioral changes, hyperactivity, anxiety, and memory deficits, and there is some kind of later life apparent degeneration. We are also doing human neurons for these individuals, which we don't have any data to share with at this point. But this is, our kind of take on this idea of gene discovery, and then deep follow through to come up with hopefully novel therapeutic targets and novel therapies. So I'll stop there. I'm sorry I ran a little bit over. And thank you from the Seaver Center, and I'm looking forward to your questions.
Thanks very much, Joel. Yes, we have 2 questions for the moment. One from Matt Third. So how confident can we be that the chromatin-related risk genes are exerting their pathogenic impact during early development as opposed to later neuronal function, like synaptic genes and MECP2?
We can be about this confident. So I think it's early stages. If you look at the GERs versus the NCs, and you look at them, for example, in BrainVar, as you know, the expression of the GERs tends to be early in development, and the neuronal genes tend to be later and kind of crossing into birth and over life. So that is, one bit of evidence. It's let's just say on average, it's true. And the other interesting thing, and again, it's a small n, is what I mentioned, that the GERs with known targets do not seem to be regulating the NC genes, but they do regulate the GERs without a known target. So we have strong p-values showing GERs regulating other ASD GERs, but we see no evidence of GERs regulating the NCs. Again, it's on average, and it's certainly not going to be true for every gene.
Thank you. We have a question from Anders Berglund. Great talk. Thanks, Joe. Let me assist bringing more calls to Newcastle. Evidence, especially from somatic disorders, suggests that stratification of patients can be improved by combining common and rare variants. Might this also be the case for neurodevelopmental disorders? And is there any potential clinical utility?
Yeah, so I think Matt showed you some of the common variation in the DDD, and Mark also showed you, some of the genes have both common and rare variation together. Bernie Devlin and his team have done a lot of work in this area, showing that what we kind of would expect, that the most deleterious mutations may have less polygenic risk, but as you go down, as the genes are less penetrant, and Mark Daly showed this as well, you see more polygenic risk. So I think the next big thing will be integrating polygenic risk and rare variant risk kind of in a joint model. And I think it will at that point have clinical utility. It may also be it may also help us understand these non-specific syndromes, because it might be the polygenic risk that's producing a more autism phenotype or a more schizophrenia phenotype. I don't know if that completely answered the question, Isabel, because you dropped out for a second, but tell me if I should say more.
No, yeah, I think it's, you gave a good answer. Thank you. The next question is from Jinji Yuan. Thanks a lot for the great talk. In the gene discovery of ASD using TADA plus model, do you have any assumptions regarding the weight of de novo mutation and case control mutations in the model?
Absolutely. The parameters are, we estimate the parameters for all of those things, and the parameters historically were estimated from a smaller sample than applied to a larger one. The parameters haven't really changed that much. They seem to be more or less on track. It's a Bayesian model, so it's somewhat forgiving on some things. One of the most interesting things, and I don't want to steal somebody else's thunder, is that, and you've already heard about it from Matt, we're not doing a great job with inherited variation for a bunch of reasons. One, of course, is that the signal to noise is huge, right? Those standing variation is in the population. So and the second is they tend to have smaller effect sizes. So TADA never really did a great job on chromosome X, because it does, chromosome X often, most of the risk comes from inherited recessive variation from an unaffected mother to a boy. But some new versions of some new analytical approaches by Dave Cutler are really doing a great job of getting X really kind of well defined. And Matt, I showed a slide of Matt Hurl's group doing something similar. And in there, we're doing a much better job with inherited variation, and applying the same methodology to the autosomes is actually doing a good job as well. So I think there are assumptions made, and there are parameters that you have to put in about weighting. The weighting, I think, is very, very robust. Obviously, as Mark also said, if something, if something is really, really, really selected against, it's almost certainly going to be, if not de novo, at least recent, right? So there is a reason to put in a different weighting for that versus something that's standing variation in the population. But I think our parameters are pretty clean, and we've refined them TDA has been refined 6 times at least within the ASC over the past years, and it's also been refined by other groups as well. So I think, and then we have a whole new version of something that's being called TDA, but is a little bit different, that actually does a good job with inherited variation, and including on the X chromosome.
Thank you. A last question from Prakash Devaraju. Great talk. Thank you. About precision medicine for a developmental disorder. How to intervene when the critical period of developmental changes have already taken place by the time the condition is diagnosed? Yes, great question.
Sure.
You could respond quickly.
Yeah, so very quickly, I mean, I think that is a $1 million question, but you know, as Adrian mentioned, when you turn the gene back on, MECP2 back on in mice, you get some recovery. Shank3, a beautiful paper about galloping fang, that if you turn the gene back on in young mice, you get more recovery, but even in adult mice, you get some recovery. It's been done for FMR1. It may not be universally true, but it does seem that you can get some recovery even at later ages. Does it mean earlier is not better? No, of course it's going to be best to do early. And when we have universal sequencing at birth, you know, maybe once there are effective interventions, I suspect that a child was showing any signs of delay at 6 months will be sequenced and maybe put into some of these interventions if they're shown to be very, very safe and effective. So we've got a ways to go, but we do have preclinical data that's very clear, and even some clinical data actually, that you can reverse things even after time passes.
Good. Thank you for this positive end note, and thanks again for the great talk and the audience for the nice questions. Thank you.
It was my pleasure. Thank you.
So now we have the pleasure to have Jonathan Mill from Exeter University. Yes, Jonathan, hello. Great to see you again. Sorry for you. And we can even hear you. That's fantastic.
You can hear me. Amazing. Well, I hope, fingers crossed, that this carries on. Yeah, and thanks very much for giving me the opportunity to speak again, and hopefully the internet won't break on me today. So, I don't know if you can see my slides. Are they? Brilliant.
Yes, we can. Yeah.
So, yeah, so I'll kick off. So the work in my group is really focused on understanding the causes and consequences of molecular variation in the brain and the role this plays in neurodevelopmental phenotypes and also mental health and disease and right through the life course, actually looking at neurodegenerative phenotypes. And I'll give you a kind of overview of some of the approaches we're taking to study this and also give you some insight into some of the real problems there are in doing this type of work and some of the issues there can be in making conclusions from the type of molecular data that's being generated. Let's work out the slide thing, as that works. Yes, brilliant. So, you know, we've heard already about the spectacular advances in understanding the genetic basis of these diseases. And of course, we mustn't forget that our genes operate also in the context of the environment. And there's been some really great epidemiological work, obviously highlighting some of the important early environmental risk factors for disease. But you know, how these things all come together to influence gene function in specific cell types at critical points of development, I think is still very much a black box, as shown in this cartoon. And I think a key challenge for our field is going to be really to understand what are the functional consequences of the genetic risk variants that have been widely associated with disease. Great, this is working. So my group takes an integrated functional genomics approach. Our aim is to really identify genomic pathways involved in disease. And I think one of the key take-home messages from our work is that despite the huge heterogeneity that we've heard about in the etiology of disorders like autism and schizophrenia, at a genomic level, actually, we are starting to see evidence for a fairly convergent molecular pathology. So a lot of our work is really focused on profiling gene regulation in human postmortem tissue, profiling multiple layers of gene regulation, and putting these data together using systems biology approaches. We're interested in development, but also aging, and using model systems to explore the functional consequences of the variation that we're identifying. And I guess ultimately the goal is very much to identify transcriptional networks that potentially represent novel drug targets that could be potentially probed with novel pharmaceutical agents. Okay, so this is just kind of hype. Don't worry about the details here. It's just some results from a recent genome-wide analysis of DNA methylation. This is in the prefrontal cortex from patients with schizophrenia compared to matched controls. What you'll see here are two modules of co-methylated loci, and these are strongly associated with disease. And these modules are enriched for differential methylation in the vicinity of genes that are very strongly involved in neurodevelopment. They're actually linked together by epigenetic variation in the promoter of the SHANK2 gene, which, as you know, is an important synaptic scaffold protein, has an important role in neurodevelopment, and has been widely implicated in psychiatric disease. It's also worth noting that, amongst these differentially methylated sites, the genes annotated to these sites, we see really strong enrichment for genes that have been implicated from genetic studies of schizophrenia. And I think this, going back to my point just now, this highlights the power of these studies in relatively small numbers of post-mortem tissue samples that are available for study, for actually highlighting important genes and pathways that have been highlighted in much bigger studies of hundreds of thousands of people in GWAS. A similar story in autism. Here we've been collaborating with Dan Gesherin's group, for instance, looking at integrating DNA modifications, markers of active enhancers, and gene expression in different brain regions of individuals who have died with autism. Of note, again, we see very common signatures across autism cases and evidence for convergent signatures between individuals with idiopathic and syndromic forms of autism. And again, the genes annotated to these differentially regulated sites are really strongly enriched for various things, immune and synaptic pathways, but also a real focus on neurodevelopmental pathways. So actually relatively little is known about gene regulation occurring during human brain development. Much of what we know comes from studies of mice and other model organisms. And of course, there are clear differences in how a mouse brain develops compared to a human brain. So we've been working with samples from the Human Developmental Biology resource and the MRC Brain Bank network in the UK to start to put together trajectories of gene regulation taking place across human brain development, integrating different levels of gene regulation. So you can see in the bottom right, this is just examples of very profound changes in DNA methylation at 4 sites in the genome across the first and second trimester of human brain development, just highlighting the really strong differences we see. Again, these changes don't just occur singly at individual positions. We see kind of whole networks of co-methylation occurring across the genome, with whole networks of genes becoming either methylated to unmethylated or vice versa. Okay, and one of the interesting things is that we see, as you might expect, widespread differences between males and females and patterns of gene regulation. But this is not just on the sex chromosomes, also on the autosomes. And perhaps, even more interesting are these examples of sites that show sex-specific trajectories of gene regulation. So on the left is an example of a site that's very differentially methylated between males and females very early in brain development, and those patterns are converging towards the point of birth, and on the right is an example where actually the patterns are diverging. And of course, this is interesting, perhaps in understanding some of the sex differences in neurodevelopmental phenotypes. And just to kind of highlight, how this relates to some of the data that we're generating on adults who've died with disease. So each column in this quite complex figure represents a site in the genome that we find differentially methylated in prefrontal cortex in individuals with schizophrenia. And on the y-axis is essentially an indicator of how strongly that site changes across brain development. And essentially what we see is a very striking enrichment for these neurodevelopmentally dynamic sites amongst those positions in the genome that we find differentially regulated in adults with schizophrenia. So here's just one example. Here, this is a DNA methylation site in the promoter of the NY1TL gene, a potent transcription factor involved in neurodevelopment. On the left are the case control differences in terms of DNA methylation, and on the right are the patterns of DNA at that same exact site across human brain development. And just to kind of highlight one utility of these types of annotations that we're generating, it's in actually interpreting perhaps the consequences of mutations that are associated with neurodevelopmental phenotypes. So here's just two examples of genes that we know have mutations or structural variants associated with neurodevelopmental phenotypes that harbor what we describe as neurodevelopmentally dynamic regulatory domains. And in total, if you take the list of genes in the Safari database, for instance, they're highly enriched for these neurodevelopmentally active patterns of gene regulation. So of course, very few of the common genetic variants that have been identified in GWAS actually bring about or exert an effect via change in protein structures, as we've heard about. And instead, these are really hypothesized to influence more subtly patterns of gene regulation. Another issue, I guess, with some of the findings from GWAS has been alluded to in the studies earlier, is that the lead SNPs or the lead variants often lie in very large regions of linkage disequilibrium that maybe contain many genes, and actually prioritizing causal genes can be a major challenge that's obviously very important to do. And one reason why it's so important, I think, to put together these regulatory genomic annotations that have been derived in relevant cell types at the relevant points in development, together with the genetic data, is it allows us to start generating hypotheses about function that can be obviously subsequently tested in functional studies. So as I said, we know that GWAS variants are enriched in regulatory elements in the genome. So this is just some data from our group where we've used LD score regression to highlight that there's an enrichment of GWAS heritability amongst, in this case, H3K27 acetylation peaks in the cortex. These are regions that are defined as active enhancers or promoters. And you can see actually this is fairly generic across all the GWAS data sets for brain disorders that we tested. What is interesting is that these enrichments tend to be very specific for brain traits. So those are highlighted in light blue in the middle. And actually, if you look at GWAS stats from non-brain traits and the white bars in the right, you'll see much lower enrichment there. And actually, the ones in the dark blue on the left are non-brain phenotypes that you could argue actually have some involvement of the brain in their underlying phenotype, things like obesity and BMI. again, this highlights the importance, I think, of generating these regulatory genomic maps in the right cell and tissue type. So the other thing in relation to integrating genetic and regulatory genomic marks is that, in fact, inter-individual variation in epigenetic regulation is under fairly strong genetic control. And we know that from studies like this. This is a study we published a couple of years ago looking at DNA methylation across the genome within a large population of monozygotic twins and dizygotic twins. And you can apply kind of standard quantitative genetic approaches to these data to essentially show that concordance rates in MZ twins are much stronger than they are amongst DZ twins for many sites in the genome. So there's an example there at the top, and you can then kind of use structural equation modeling to work out the contribution of genetic and non-genetic factors to variation at that site. And what we find is, of course, that those that are under very strong genetic control, we can then do, essentially do GWAS of DNA methylation and identify these DNA methylation quantitative trait loci, as shown in the bottom, where different genotype groups associate with different levels of DNA methylation at a specific site. It's worth noting that, not all variation is driven by genetics in terms of DNA methylations. There on the bottom is an example of a site that's highly variable, but equally variable in monozygotic and dizygotic twin pairs. And these might be sites that are potentially environmentally sensitive. And my clicker's stopped working. Oops. Okay, so we can start. Hopefully that's visible. So we can start mapping these genetic effects on DNA methylation and other regulatory marks across the genome in relevant cells and tissue types. And so we've generated maps, MQTL maps in fetal brain. That's just an example shown here. And we find, as you might expect, that most of these associations occur in cis, so that's genetic variants influencing DNA methylation quite locally. But we do see quite a lot of evidence of trans effects. I think these are particularly interesting because they tell us that maybe the functional consequences of genetic risk variants aren't necessarily local to that variant, and actually we should be looking in terms of genic annotations at more distal sites. The other thing, and you see in the heat map at the bottom, is when you look at effect sizes at these QTLs across different brain regions and different stages of development, actually these genetic effects tend to be fairly stable. But I always think the exceptions to the rule are really interesting. And so here's an example of a DNA methylation QTL that's fetal cortex specific. You'll see that in the red bars and the left. Each column is a different genotype group. You'll see a strong effect in fetal cortex, no effect in adult cortex, adult striatum, or adult cerebellum. And here's an example where actually we see an opposite effect in terms of gene regulation in fetal cortex compared to, in this case, the adult cerebellum, where the genetic variants are actually having an opposite effect on DNA methylation. And we took these fetal cortex MQTLs, and because we are interested in the hypothesis that schizophrenia is a neurodevelopmental disorder and the genetic variants may be influencing their effect during brain development, we were able to show that the schizophrenia risk variants are very strongly enriched for fetal cortex MQTLs. And actually, if you then take, this is specific to schizophrenia, because if you take equally powered GWAS from, say, Alzheimer's disease, which is obviously a disease of neurodegeneration, and non-brain related GWAS data sets, you don't see the same enrichment. And we see very similar for EQTLs as well, generated in fetal brain. This is work in collaboration with Nick Bray in Cardiff. So we can, once we've generated these databases of QTLs, we can actually start to use them to help fine map GWAS regions and prioritize genes for more targeted investigation. So here's a GWAS region from the schizophrenia GWAS, and as you'll see, the problem here is that this is a big LD block, and there are 5, 6, 7 genes in this LD block. So, which is the gene that we take forward for our functional work. And we're able to use approaches like Bayesian co-localization approaches to identify variants that are associated with both disease and genomic regulation, and then take these forward for the downstream work we're doing in terms of characterizing these functionally and doing various model work on them. Another similar type approach is using summary database Mendelian randomization to identify pleiotropic relationships between disease and markers of gene regulation, allowing us to annotate GWAS variants to target genes in specific tissues and cell types. And just as a bit of an advert, this is a data set that people may be interested in. This is the work of the Genetics of DNA Methylation Consortium, which has just undertaken the largest study of genetic effects on DNA methylation. This is work led by Josie Min at the University of Bristol. It's essentially a big GWAS of DNA methylation in 30,000 individuals. Take-home message is that we find that almost half of DNA methylation sites we assessed are actually at some level under genetic control. And you can use this data to start making biological insights about disease traits. All the data is available online, and the link to the preprint is up there if people are interested. Okay, so now that we've started to define these epigenetic marks across neurodevelopment, we want to start to explore what the functional consequences of the variation is what the functional consequences of the variation is. And to cut a very long story short, we find real interest in the area of alternative splicing. It looks like a lot of the regulatory variation that we see is influencing isoform diversity and splicing in the brain. And we've heard already from some of the short talks the importance of alternative splicing in terms of development. It influences many, almost every human gene has alternative splicing or different isoforms. We know it's involved in development, and importantly, these different isoforms have different functional effects, which are obviously potentially important. Now, one of the issues with standard RNA sequencing is that it's very hard to actually really define define transcript variation, because the reads are so short, and you have to rely on these small number of reads that actually span splice junctions. So we're leveraging the power of long read sequencing, specifically by Pacific Biosciences and Oxford Nanopore technology, to generate novel transcript maps of the human and mouse cortex and the cross development. Again, I'll just touch on a few highlights, but all our data is freely available. Please email me if you want the link, and there's a preprint down the bottom there. So this is just an example, just to highlight the amazing power of these long read sequencing approaches. This is the most isoformic gene we found, which is MEG3. It's a maternally expressed imprinted long non-coding RNA that's involved in synaptic plasticity. And we found all sorts of novel forms of this gene that aren't in any existing gen code annotations. That's one of the things here. Over 40% of the transcripts that we identified in this work are novel. This is TCF4, obviously well known to people working in schizophrenia. And this cartoon here just gives one example of a gene that really highlights many of the different types of alternative splicing and transcript variation that we see. So we see examples of exon skipping, intron retention, alternative first exons. And particularly interesting to me are these 1% of transcripts that represent fusions actually between adjacent genes. And we found that these were actually fairly abundant in genes implicated in neurodevelopmental phenotypes. So on the top there of interest to this conference is a fusion between MECP2 and the adjacent gene IRAK1. But we found these to be pretty prevalent amongst genes that have been implicated in both autism and schizophrenia. And as I said, we're validating these and looking at proteomic effects. We see differences across between fetal brain and adult brain. I'll skip the details here. if you're interested, read that paper. So we're also interested in the life course approach, and what I've talked about so far are changes and differences in fetal brain development, but actually we're now assessing changes that are taking place in gene regulation across the life course. You'll have probably heard a lot about epigenetic clocks and interests in these. And one of the problems on those is that they've all been calibrated in blood, and actually they don't work very well in the brain. So if people are interested in brain epigenetic clocks, we've just published a DNA methylation clock of the human cortex that actually works really well and enables you to start looking for accelerated aging. I'll just highlight this because I think it's a really important gap in our knowledge. So this cartoon here kind of highlights all the samples we're looking at and different modules of gene regulation that define different brain regions across time. But there's this whole window of the third trimester and early neonatal period that we know very little about. And we've just got a grant from the Simons Foundation to start really filling in that gap, really. I think that's going to be a really important window into what's going on. And just to highlight, how differences later in life can really lead to important neuropathological differences. This is our work on Alzheimer's disease that's highlighted really robust differences in DNA methylation associated with Alzheimer's disease. And these are the reason I flagged this up is that these effects are very specific to the region of the brain that's implicated in Alzheimer's disease. They're pretty much only seen in the areas of the entorhinal cortex that are affected very early on in neurodegeneration. And because we're using older samples here, we're able to leverage the fact we can collect pre-mortem blood samples from the same individuals. And the bottom line is that we don't see any evidence of the changes that we see in the disease-relevant tissue in peripheral blood from the same individuals. So this brings me on to really the final bit of the talk. I just wanted to highlight some of the issues of doing this type of work. And I guess this applies to molecular epidemiology in general and not just epigenetics. And that's the fact that if you're doing genetics, life is relatively easy because all you need is a blood sample. Every cell in your body has pretty much the same DNA sequence. Obviously, there are some rare somatic mutations. It doesn't matter when you take that DNA sample, you are born with the gene sequence that you die with. It doesn't matter what you do in your life, it won't change your DNA sequence. The disease itself won't affect your DNA sequence. And obviously there are really great reference genomes that we can relate that to. And it's really worth remembering none of this is true in the context of epigenetic epidemiology. And I just want to talk through an example from our work to highlight where things can go really wrong, so I'm not embarrassing anyone else. So this is a study of DNA methylation in schizophrenia cases compared to controls. It's a big study for an EWOS analysis, and you get Manhattan plots just like you would in a GWAS, and you can start getting very excited and tell stories about the genes that are annotated to the sites that you find to be differentially methylated. But actually, if you look at that Manhattan plot and compare it to this, you'll see that they're virtually identical. And this is actually a Manhattan plot from an epigenetic study of cigarette smoking. And of course, epidemiology tells us that smoking is much, much higher in schizophrenics than it is in the general population. And essentially what we had done is a study of smoking and not schizophrenia. So really worth remembering when reading or doing this type of work that designs that have been really optimal for genetic epidemiology are not optimal for molecular epidemiology. Bigger isn't necessarily better, and you can't just run methylation assays on samples collected for GWAS. There is a flip side to that, and actually you can gain some really useful information from this methylation data. So actually with the smoking associations are so strong, you can actually derive algorithms that allow you to work out not just who is a smoker, but how much they've smoked and how long they've been smoking for. And that works really well. If I was to take a DNA sample from any of you, I could tell if you were a smoker or not. And just to show a real life example of how this is applied, this is data from our collaboration through the Minerva project with iPsych, looking at DNA methylation in neonatal blood spots, and then stratifying those samples based on this smoking score, whether the mother smoked during pregnancy or not. And you'll see that even, you can see these very robust signatures of smoking in, blood spots from individuals, a couple of days old, based on whether their mum smoked or not during pregnancy. So I think that's not only an important public health message, but it also highlights the huge potential for confounding in molecular epidemiological studies of disease. Individuals on with schizophrenia are obviously on long-term treatments. This is just longitudinal DNA methylation data from clozapine from treatment-resistant schizophrenics who are naive from clozapine at baseline here, and then we've tracked their methylation across their treatment regime, and you can see again very discrete patterns of changes. And again, we can actually derive these scores that actually allow us to measure how much exposure to clozapine an individual has had based on their DNA methylation levels. If people are interested in some of this work, this paper's just out in eLife, which allows you, which documents some of our schizophrenia biomarker work in terms of methylation. So the other big confounder in this work, and this is the final thing I'm going to talk about, is cellular heterogeneity. And I think I've probably alluded to this, but obviously each tissue is made up of many different cell types, and each of those cell types has a very different epigenome, and the proportions of those cell types can actually differ as a function of things that relate to disease. So again, huge potential for confounding. So we've spent the last kind of 2-3 years really refining a method that allows us to take a lump of cortex tissue from this very valuable resources that we have access to from people who've donated their brains. Obviously, we want to get as much information from each individual brain donation that we can. And so we're able to use markers for neurons, oligodendrocytes, microglia, and other cell types, all in parallel to purify pools of nuclei from frozen tissue. We can validate that this has worked using single nuclei RNA-seq, and then use these pools of nuclei for downstream genomic profiling. And just to show you that this works really well, this is across the MOG gene expressed in oligodendrocytes. In the pink, in the purple is the neuronal profile, and the pink is a oligodendrocyte profile. And you can see very big differences between those cell populations for the different marks of gene regulation that are expressed here. And,
yes,
from these we can derive statistical deconvolution approaches that can then be applied to bulk tissue. And these really do work. So this is just showing differences in neuronal proportions between our Alzheimer's disease cases and controls. But a much more powerful approach is actually to purify these populations across many individuals, disease cases versus controls across development, and actually start defining epigenetic variation within across individuals within each of these purified cell populations. And this is now really the kind of route that our work and the work of others is taking. And I just want to leave you with a teaser of some work from a big study of schizophrenia we're doing, looking at about 300 brain samples, purifying these 4 different cell populations individual, and starting to identify cell type specific associations that would be hidden or masked totally if we were simply profiling bulk cortical tissue. So this is my concluding slide. I think, using these types of marks really does have a future for informing not just etiological studies, but also the future in terms of personalized medicine. I think we're starting to uncover epigenetic biomarkers of disease and exposure. We're learning about mechanisms of disease. We're refining traditional risk models. We're looking and identifying biomarkers of drug response, but ultimately in the future, because these modifications are potentially modifiable with pharmaceutical agents, they provide, I think, really great targets for future and therapeutic intervention. So the most important slide here is the acknowledgements of my team and the people who fund our work. Please get in touch if you want to learn more about any of this or want to access any of our data. We're big proponents of data sharing, and I'd love people to use some of our resources that we're generating. Thank you.
Thanks very much, Jonathan, for this super informative talk, and also for the note of caution about data and methylome profiling and the issue of cellular heterogeneity. I think it's just stress the necessity for single cell analysis that many labs and many people are indeed implementing in their research. We have a question from Flora Vaccarino. Were you able to identify relationships between patterns of methylation in the developing brain and transcription? And is there a pattern of methylation that might identify enhancers or repressors?
Yeah, so we're certainly trying to tackle that, and I don't know if the question was posed before I got to the bit on isoforms, but so the actual direct relationship, for instance, between the DNA methylation signatures and the gene expression changes that we see using sort of standard RNA-seq aren't necessarily as clear-cut as you might expect. That I think that direct relationship between levels of gene expression and epigenetic status, isn't like you read in the textbook where it's switching genes on or off. As I said, the real interest, I think, is actually, well, a large amount of the interest from our perspective is in how it's regulating the different usage of exons and different versions of transcripts, different isoforms at different stages in development. And I didn't have time to dwell on some of the differences we see between fetal cortex and adult cortex in terms of transcript usage, but they're really, really quite dramatic. So the overall level of gene expression, maybe, if you were just doing a sort of standard, how much of gene X is there, would look fairly similar, but the structure of the transcripts that are being made in many cases are quite dramatically different.
Okay, thank you. A question from Cloud Battu. Super exciting, thank you. When do you plan releasing your single nuclei schizophrenia study?
Yeah, well, it's work in progress. So we've, as I said, that getting the assay working, so the sorting the nuclei populations has been really difficult. So it's quite straightforward to do it for a population at a time, but what we were really keen to do was multiplex as many different cell markers as we could into one FACS sort, and then generate from one lump of 500 mgs of tissue enough pools of nuclei of each of the different cell fractions in order to do all the different molecular assays we wanted to do. So we're now, in terms of our schizophrenia project, we're generating data. We've probably got through about 150 brains at the moment. You can only do 2 brains a day, so it's pretty laborious work. But so we have our initial results. I'm really happy, as I said, to share things and for people to look up things in our data. So that's not single cell data. That's basically lots of cells, but purified populations of cells. We are in parallel doing some kind of more traditional, I guess, 10x single nuclei RNA-seq and single nuclei attack-seq. And again, we're at the stage of data generation, but if it helps anyone and that data is useful, we'd more than happily share it.
Good. We have a last question from Chan. You mentioned MEC3 is involved in synaptic plasticity. In which aspect can you elaborate a little bit more? Maybe quickly, Jonathan.
So someone else can elaborate. So that's me reading up on how MEC3 is involved in the brain. So I won't take credit for saying that's involved in synaptic plasticity, but and I don't know off the top of my head the reference I was citing at that point, but the internet tells me that MEG3 is important in synaptic plasticity. So there you go.
Very good. Well, thanks very much for this super enlightening talk, and thanks to the audience for the questions. So this closes the afternoon. So thanks again to all the speakers. And I also would like to thank the Lundbeck Foundation, in particular Martin Meyer from the foundation, and also Mats Sorensen from DVC company, the company which took care of the online meeting. So they did a great job in preparing the speakers and the chairs to make this meeting run smoothly. So many thanks. And tomorrow, so the third day of the meeting, will be on neuronal dysfunction in neurodevelopmental disorders, and the afternoon will be chaired by Konstantin Kodosevich. And among the speakers, there will be Claudia Bani from the University of Lausanne talking about motor and social skill development. Jessica Cardin from Yale University talking about GABAergic contributions to neurodevelopmental disorders. And Flora Vaccarino also from Yale, talking about cell-fate trajectories in cortical organoids from autism spectrum disorders. And there will also be, like today and yesterday, flash talks by 3 people. So many thanks again, and I wish you a nice evening and nice afternoon, wherever you are. Thank you, and bye-bye.